Hello students, welcome to Aakash. I'm your botany faculty, Dr. Sukirti, and today we are here to discuss the chapter molecular basis of inheritance, right? And as you know, it is a very important chapter with respect to NEET, because every year like 5 to 6 questions coming from this particular chapter, right? So that's why we are starting this chapter, and we'll try to cover all the aspects of this chapter in this particular session, right? So let's begin. Good morning everyone. So we'll begin with central dogma of life, right? So what basically the central dogma of life is, that it is a particular set of principles that are incontrovertibly true, right? तो ये हमें actually बताता है that how exactly the life forms run its processes, right? So this particular dogma starts with the genetic material, which is DNA, right? Now when it makes its copies, then that process is known as replication. Then DNA convert itself into, not convert exactly, but can form mRNA, right? So this process is known as transcription. And then further, when this mRNA is used for the synthesis of protein, that process is known as translation, right? So major chunk जो है इस chapter का, it is going to be driven by this central dogma only, ठीक है? We'll start with the very first molecule, DNA itself, right? क्योंकि हमें इसी particular molecule से start करना है. That is the reason first thing that we need to know is that what this molecule is all about, right? So when we talk about DNA, so initially Friedrich Miescher has discovered an acidic substance from the nucleus, and he said that particular thing as nuclein, right? Now people are confused that what is nuclein. So Altman has performed the basic characterization and gave the term nucleic acid, right? तो हम जानते हैं that DNA का full form क्या है? Deoxyribonucleic acid, right? So this is how nucleic acid term came into existence, right? So when we talk about this DNA, so this is a polymer of nucleotides, right? So we'll begin with firstly what is a nucleotide, right? So if we talk about the structure of a nucleotide, then we'll see certain important thing. Like the very first thing over here is this pentose sugar, right? 5 carbon sugar, that is a characteristic sugar that's being found in the nucleic acid. Whether we call it as DNA or we call it as RNA, दोनों में ये pentose sugar common है, ribose sugar common है. The only difference between this sugar, RNA और DNA में क्या difference है? अभी जो sugar है, अगर let's suppose हम इसे name करते हैं 1 prime 2 prime, right? आपने organic chemistry में ऐसा किया भी होगा. So अगर आप देखो यहाँ पे 2 prime जो carbon है, उस पे still OH is being present. So this particular sugar is the ribose sugar, right? और ये sugar हमें किस में मिलती है? RNA में. In opposed to DNA, DNA में क्या होता है? This oxygen is not present, right? And that's why this particular form of sugar is deoxy. D means absent. What is absent? Oxygen has been absent. So deoxyribose sugar, and this particular sugar is being found in the monomeric units of DNA, right? Now the second important component of a nucleotide is a nitrogenous base, right? So this nitrogenous base can be of 2 types, of course. It could be purine, right, like adenine and guanine, and it could be pyrimidine, cytosine, uracil and thymine. So we know कि A and G, right, these two are being common for both RNA and DNA. But when we talk about the pyrimidines, cytosine is common in both DNA and RNA, but this is specific for RNA and this is specific for DNA, right? So these are the nitrogenous bases. Now when only pentose sugar and nitrogenous base are together, like only these two are together, then we call this complex as nucleoside, right? And when, now how these two are going to be together? They have to make a bond with each other. So this particular bond that they are having with each other is known as N-glycosidic linkage, right? Because it's been attached with an N atom. You have seen O-glycosidic bonds when you have studied the carbohydrates in the biomolecule chapter, right? So over here we'll be finding N-glycosidic linkage. Now, when the nucleoside is being attached with a phosphate group, then it is going to form the nucleotide. And the bond over here, right, it's been connected to the 5' carbon that's being found in the ribose sugar. So when it is this phosphate group gets connected with the sugar, this particular bond is known as phosphoester bond. Do remember, it's not phosphodiester, it's still a ester bond, right? ROR, right? This is an R group. So phosphoester, because one side you will be having phosphate group in opposed to this. So this is RO and phosphate group. So that's why phosphoester bond, right? So this is what a nucleotide structure looks like. This is a monomeric unit of the DNA or RNA, right? Now, in order to make DNA and RNA, these nucleotide needs to be, combined with each other. So for that, this is how the bonding occurs. So the 5', this phosphate group which is present at the 5', it is going to interact with the 3' OH group of another nucleotide, and that's how there will be a creation of phosphodiester bond, right? Why we are calling it as diester? Because one is this R, O and this phosphate, and then agar idhar se dekhe, so this one phosphate and then this O and then this R. So this is 2 times ester bond formation is occurring. So that is the reason why we are calling it as phosphodiester bond, right? And when multiple nucleotides are going to be added up together, we'll be getting a structure like this, the structure of DNA, right? So was it this simple? Was it like this is the only thing by which we have devised the entire structure of DNA? No. There are scientists who have given, paramount contributions in order to decipher the structure of DNA, right? And those 2 major contributions are of, first, Maurice Wilkins and Rosalind Franklin. Right? So these 2 have given the X-ray crystallographic data, the X-ray crystallographic imaging the X-ray crystallographic data, and this data suggested that DNA is going to be present in a helical form, right? And it is showing repetitions at a particular interval, right? Like after every 3.4 angstrom, there is a repetitiveness that we could see, right? So this is their contribution. They are not able to decipher the exact structure. They have given certain points regarding the structure, that firstly it is a helix, and then we can see the periodicity in its structure. Then the second major contribution was of Erwin Chargaff who said that the amount of purine is going to be equal to the amount of pyrimidine. But there is one important thing to notice here is that Erwin Chargaff, whichever propositions he have proposed, is for double-stranded DNA, right? It is not applicable for single-stranded DNA or RNA, right? It is only applicable for double-stranded DNA. So in case with this, you must have solved various, numericals in which, let's suppose, percentage of guanine has been given and you have been told to, enumerate the percentage of adenine or maybe cytosine or thymine. And let's suppose it has been mentioned in the question that it is for single-stranded DNA. Then you cannot apply this particular rule over there, right? So Erwin Chargaff proposed that purine and pyrimidine is going to be present in the equal amount, right? So purine means A plus G and pyrimidine means T plus C, right? So this proportion is going to be equal, or we could say A plus G upon T plus C is equal to 1 is going to be the constant for all the species. Thus ratio that is going to vary for the species would be A plus T upon G plus C, right? This ratio, this is known as base ratio. This is going to be changed for the species, right? So these are the propositions that has been helpful for the deciphering of the exact structure of DNA. So both of these particular works were not able to give the exact structure. Now the exact structure was given by Watson and Crick, right? These two have given the double helical model of DNA, which is being considered as the most appropriate demonstration of how the DNA structure looks like, and that is the reason why they have got the Nobel Prize as well in 1953, right? So according to their idea of Watson, according to their idea of double helical model, firstly they proposed that the DNA is going to consist of two strands, which you could clearly see over here, right? This is one strand and this is another one, right? The second thing they have proposed is that the backbone, that means these strands, are going to be made up of sugar and phosphate. So the backbone is going to be of sugar and phosphate. This is first proposition. Then the next one is that the two strands that are being present are going to be anti-parallel to each other. Anti-parallel means if one strand is going to have the polarity of 5' to 3', then another strand is going to have the polarity of 3' to 5'. 3' is the end where an OH group is free. 5' is the end where a phosphate group is free. So both of them are going to have the opposite polarity, anti-parallel in nature. Another thing that they have proposed during their model is that the nucleotide, the nitrogenous bases that we are going to see, will be present towards the inner side and they form hydrogen bonds with each other, right? Now this is one of the important hallmarks of this particular model, that hydrogen bonding occurs between purine and pyrimidine. So according to their model, they have proposed that adenine is going to pair up with thymine with the help of two hydrogen bonds, while cytosine is going to pair up with guanine with the help of 3 hydrogen bond, right? They are purines, the heterocyclic rings, right? Derivative of purines, no, they are the purine structures. Derivative of purines, you can see PGRs, like if you see cytokinin, that is a derivative of purine, right? So this is again one of the hallmarks of the double helical model of DNA, that they have provided the base pairing between the nitrogenous bases. Right. Now, apart from that, another thing that they have proposed is that the pitch of the DNA is going to be 3.4 nanometer, right? This one particular turn that it took is of 3.4 nanometer. And in this one turn, we are going to have 10 base pairs approximately, right? Then the distance between two base pairs, like this is one base pair, T is pairing with A and another is C is pairing with G. So this particular distance between the two base pairs is 3.4 nanometer, 0.34 nanometer, right? It's not 3.4. If I'm saying that it is 3.4, then the unit will be changed. It will be angstrom, right? Similar over here, if I say the pitch is 3.4 nanometer, I can also say that it is 34 angstrom. So do see the units while answering the question, that what's the pitch of the DNA and what is the distance between the two base pairs, right? So according to this particular, model of DNA that has been proposed by Watson and Crick, they have said that DNA is a stable structure, and two things are basically, conferring this particular stability. One is the presence of this hydrogen bond, and second is the stacking of base pairs. Over here, one base pair is going to stack over the other like this, right? So this stacking is providing the extra stability to this particular structure. So this is how Watson and Crick have proposed the model. So this is the first contribution. Another contribution of Crick was the proposition of central dogma itself, right? So he has given too many contributions, and we are going to see the contributions as we are going to progress with the chapter. But two of the contributions we have studied just now. One is the proposition of this double helical model of DNA, and another is central dogma, right? So this particular model has given the exact data that how exactly the DNA structure looks like. Okay. Now the next thing that comes into mind is that how this particular DNA is going to fit into the nucleus, because if we see the length of the DNA, it's too huge, almost 2 to 2.2 meters. So this much long DNA, how are you going to accommodate it in a nucleus which is so small? So for that, what we require is DNA packaging, right? So it's not like that the packaging is going to occur only in eukaryotes. It occurs in prokaryotes as well, right? It's not like that because prokaryotes does not have a nucleus, so that is the reason why कि उनका जो DNA है वो scattered रहता है throughout the cell. That's not the case. Their DNA is being concentrated at one particular region, and that region is known as nucleoid, right? And how they are able to concentrate their DNA into that one specific region, it is by the process of packaging, right? So in case of prokaryotes, if we see, right, so we know for a fact that DNA is going to be negatively charged. Now why DNA is negatively charged? It is because of the presence of this phosphate group, right? This phosphate group being negatively charged, the overall charge that we are going to get on the DNA would be negative charge, right? Now if you want to make this structure condensed, what you require is a positively charged entity, right? Over that only you can wound it up properly. So that positive charged entities are proteins, right? In case of prokaryotes, this structure is polyamines. So these are the structure that will help to make this long DNA constricted into a small space. It's like DNA is a thread and we are going to wrap it around a small, protein or polyamine structure, right? Now, the prokaryotic packaging is not like detailed explanation. We don't have a detailed explanation. So the questions also doesn't come up from this particular, part. Major part comes from the eukaryotic packaging, right? So if we talk about the eukaryotic packaging, so in eukaryotic packaging, again of course DNA is going to be negatively charged, so it needs a positively charged structure so that it could be wounded upon it, right? So this is DNA, if I talk about, so this DNA which is negatively charged is wrapped around the positively charged histone octamer. Right? So this histone octamer Octamer means there will be 8 subunits that's going to be there. So this is how, these are the like behind structures. So exactly this is how a, octamer is going to be formed. So why this particular histone octamer is positively charged, right? So this is being positively charged because of the abundance of lysine and arginine type of amino acids, which are positively charged amino acids. Because of the abundance of those particular amino acids in these histone proteins, this overall octamer is going to have positive charge. So the negatively charged DNA can easily wrapped across this particular positively charged histone octamer, right? So when this particular, binding is going to occur, like this wrapping around is going to occur, this structure which is going to be formed is termed as nucleosome. This is the first set of what you could say condensation of DNA so that it can be accommodated in the nucleus. So over here when DNA is going to be wrapped around, it is going to form nucleosomes. Now further, the linker DNA, this is going to be attached, like you can see over here, these ball-like structures having the thread-like structure wrapped around them, and then these are the yellow kind of structures that are being present are the H1 linker histone. They will further allow the DNA to be condensed, right? Now one important point before we go ahead is that in NCERT it's been specifically mentioned that whenever the DNA is going to wrap around this histone octamer, this wrapping is going to have almost 200 base pairs, right? So this is really important because certain times you may get the questions in which they'll ask you to find out the number of nucleosomes. So they'll give you that how many base pairs are going to be present, and then they'll ask you that how many nucleosomes are going to be there. Like for example, if they say that there are 400 base pairs that are being present, then how many nucleosomes are going to be there? So you simply have to divide it with 200 because in one nucleosome, the number of base pairs of DNA that's been included are 200, right? Now, so is this particular nucleosome is capable enough to constrict the DNA into that small nucleus? No. Further compaction is still required, right? Because whenever you must have seen, the DNA in the microscope, you must have seen it either in the form of chromosome or in the form of chromatin, right? So this nucleosomic structure is basically, forms the beads on string type of appearance, right? Which we see in the interphasic nucleus when we see it under the microscope. So that particular structure, the bead on string structure, which we call as chromatin, is being formed by the nucleosome. So we can say that nucleosome forms the repeating unit of chromatin structure, right? This is really important. Now, this much condensation is not enough. We have to make the condensation done till this particular stage, the chromosome, right? So this is being done till the, you could say, the interphasic, nucleus. Then further the compaction occurs, and further compaction doesn't require histone proteins. On the contrary, they require non-histone chromosomal protein, right? Anything which is beyond this linker histone level, right? So further compaction when is being done, this particular structure is known as solenoid structure, right? It will reduce the length of the DNA and increase the width to 30 nanometer. Further, the compaction is going to occur like this, right? This is super solenoid, this particular structure. Further compaction is super solenoids, right? And now if you could see, the length has been reduced, but the width has been increased to 300 nanometers. Now further compaction is going to occur, 700 nanometer, and finally we are going to condense it till the stage of chromosomes, right, which we could see in prophase and metaphase. Metaphase, the condensation is complete. So this particular structure we are going to see in the metaphasic, cell. Now this is the microscopic image that you see actually. But what we can see that it is not that simple to make. It requires a lot of packaging around, only then you can make this particular structure. And this is how you compact the very, lengthy DNA into a small nucleus, right? Now, after this particular packaging, we know that we have created two types of, chromatin structures. One is heterochromatin, another is euchromatin. Now, what is the difference between the two? Firstly, the heterochromatin, I am going to write it as an HC, and euchromatin as EC, right? So, if we talk about heterochromatin, so it is tightly packed structure. And this is loosely packed. So how could you see it in the chromosome? So if you will see regions like centromere, right, highly constricted regions, highly, coiled ones. So that is representing heterochromatin. On the contrary, if you will see in between the chromatids, you will see the euchromatin part. Now, because it's really tightly packed, that is the reason if you will stain it, it will be darkly stained. On the contrary, if you see euchromatin, which is loosely packed, it is going to stain light. So this is lightly stained. Now, is it the only relevance of these two structures? No. The relevance of these two structures, why we are actually studying them, is because heterochromatins are the regions which cannot be transcribed, right? So they are transcriptionally inactive. And if we see the euchromatin region, they are transcriptionally active. Right? This is inactive, not active, right? So this is how the difference is going to be created between heterochromatin and euchromatin, because they are transcriptionally inactive. That means they are not also going to produce proteins for the functioning of the body. These particular structures are transcriptionally active. That means further translation process is also possible with them, and they are going to make or encode themselves or express themselves in the form of proteins, right? So this is how the packaging is being done. Now, now the question comes, was it that easy that we have started the central dogma with DNA itself, right? So, initially if you see, now we know, after you have already studied the chapter biomolecules, you must have studied that what is the importance of carbohydrate. It is the metabolic fuel, right? It could be used for generating the energy for all the metabolic processes. If you see proteins, majority of the structures in the body are being made by it, right? You see lipids, it forms the plasma membrane, the very, delimiting boundary of a cell. But at a long period of time, we were unaware that what is the importance of DNA in the nucleus, right? So, after, the Mendelian genetics have came into picture, and he said that there are certain factors which actually transmit the characters from one generation to the next. In that particular time, everyone was searching about the genetic material, that what is that genetic material that could pass from one generation to the next or from one organism to the next, right? So the very preliminary experiment, that is the transforming principle, was done by Griffith, right? In order to understand that whether there are certain structures which can transfer from one organism to the other or not. So Griffith has took the experimental material as experimental organism that he took was Streptococcus pneumoniae, right? Now this particular bacteria has got two strains. One is R strain and another one is S strain. Now the major difference between these two is, when we talk about the R strain, then this particular strain is forming the rough colonies. That means it lacks the outer polysaccharide coating, because of which the appearance of its colonies would be rough. On the contrary, if you see the S strain, right, which is the smooth strain, so if you will see, then there will be an outer boundary, an outer coating of polysaccharide, which gives it the shiny appearance or the smooth appearance when it forms the colonies, right? Now, another important difference between the two, when we talk about the R strain, because it lacks that outside covering, and that is the reason why it is being identified by the host immune system. So whenever it infects the organism, then what will happen? That the organism is going to develop the disease. On the contrary, if S strain is going to infect it, then in that particular time what happens? Because of this external polysaccharide coating, it somehow evades the immune system, and because of which it is capable to, infect the organism, right? So this is a fatal strain, while this one is the normal strain. It is not going to induce the fatal infection. Now, the test organism on which the infectivity of this particular organism needs to be tested was mice. So the test organism he took was mice. Now, what he does in his experiment is that firstly, he injected R strain, live R strain, into mice, and mice was not affected, so he was live. This is a control experiment that he has performed. Then he injected similarly S strain live into mice, but the mice got dead. And that's ensure the fact that this polysaccharide coating outside the cell is basically helping the bacteria to evade the immune system, right? Then we know that the most, crude method of killing the bacteria is to heat it, right? So he, heated these two strains to have the heat-killed strains, right? And when he injected the heat-killed strain, whether it is R strain or the S strain, into the mice was live, because the dead bacteria cannot cause the infection. But then he injected a combination. He injected S strain, which is heat-killed, and R strain, which is live, into the mice, right? So separately हमने क्या देखा था, that whenever you are going to inject the S strain heat-killed into the mice separately, then the mice is going to be live, because dead bacteria cannot cause infection. And now if R strain live is going to be independently injected into the mice was live, because of the lack of the polysaccharide coating, this R strain is not capable to cause the infection. So he thought that if the combination is going to be injected into the mice should be living. But what he observed to his surprise was that the mice was dead after the injection. So he proposed that there is certain material that has been transferred from the S strain to the R strain, which has transformed this particular R strain into S strain, right? मतलब उसके अंदर से S strain के अंदर से कोई particular molecule जो है वो transfer हुआ इस R strain के अंदर, और R strain बदल गया into the S strain, and that is the reason why the mice was dead, right? But Griffith was not able to propose that what this material is. He was not able to give the physical identity of this material, right? So the scientific fraternity wants to, identify that what particular material has been transferred from S strain to R strain. So that particular characterization of that molecule was done by Avery, MacLeod, and McCarthy, right? These three scientists have came up with the biochemical characterization of transforming principle. So firstly, what they have done, they have isolated, three things from the heat-killed S strain. And those three things are the contenders of genetic material. First was protein, right? Because it is, performing too many functions in the body itself. So initially people thought that it may be a genetic material also, right? Another contender was RNA because it's a nucleic acid, and then DNA, of course, right? So when these three have been isolated, they have been put in 3 separate test tubes, right? In one of them, so in these test tubes, there were R strain bacteria, right? The culture of R strain bacteria. So when they have put the protein, and after a while they have checked out that whether certain R bacteria has been transformed or not, so they have identified that no such transformation takes place here. Similarly, when they have introduced RNA over here in this particular, culture, again no transformation has been seen. But when they have put DNA into the test tube having the culture of R strain bacteria, they have found that certain R strain bacteria have transformed into S strain, right? So they said that DNA is the genetic material. In order to cross check it again, what they have done, that they have took the same combination, S strain heat killed plus R strain live, right? And now they have treated this particular, suspension with protease to check that whether the transformation is going to take place or not. So the transformation still happened. Still, when it has been injected to mice, the mice died. Now in the second treatment, they have used RNAs, right? They have used RNAs, enzyme, and again the transformation takes place, and that's the reason why when this particular solution along with RNAs has been put or been injected into the mice died. Then finally when they have put the DNAs into the solution, that's when the transformation stopped. And this is a profound proof that DNA is the material that has been shifted from S strain to the R strain, because of which the transformation of R strain has been done, right? So according to these three, they have proposed this proposition that DNA is the genetic material, but still the fraternity, scientific fraternity, doesn't believe them, right? There are many contradictions, and that is why they have just proposed the biochemical characterization of transforming principle by proposing the fact that it was DNA that has been transferred from the S strain to the R strain, which has caused the transformation of R strain, right? I hope till this particular point everyone is clear. In case you have any doubts, please put on the chat box so that we could discuss those doubts. All right. So after this characterization has been done, it was identified that yes, DNA is capable to move from one organism to the other. But now still the proof lacks that whether DNA is the genetic material or some other thing is the genetic material. Is it protein? Because still people are believing the fact that if protein is performing too many functions in the body, why it cannot behave as a genetic material, right? So in order to solve this dilemma, the experiments with bacteriophage came up, and this experiment was conducted by Hershey and Chase, right? So they said that we have two contenders now. One is protein and another is DNA. So the organism that we are going to use now will be having only these two components, right? And which particular organism is only having nucleic acid and protein as its constituents? So yes, that is virus, right? So they have selected a bacteriophage, right, which is having DNA as the genetic material, because till their time they don't know that they have the genetic material as DNA, but it has DNA and it is having a protein coat, right? So in order to see that which particular one is going to be the genetic material, the line was very clear that the genetic material should be able to move from one organism to the other, right? So they said that whichever molecule is going to be transferred from the virus to the bacteria would be the genetic material, right? So in order to perform this experiment, they have used two sets. In one of the sets, they have labeled the protein, right? Because protein is having only sulfur as a specific element, so that is why they have labeled it with the radioactive sulfur, which is S35, right? And in the second set, they have labeled the DNA. And now because DNA is having phosphorus as the unique element, so they have labeled the DNA with phosphorus, the radioactive phosphorus, that is P32, right? So after the labeling has been done, then they allowed this particular virus to infect the E. coli cells, that's the test organism. Now when they infected, in both the cases, he was not able to see that which particular component has moved from the virus to the bacteria, right? So after the infection, they have performed blending. Now what is blending? Blending is the mechanical, agitation. So this particular step has been done so that they can ensure that virus is still not attached with the bacteria, right? Because what exactly virus does is, or the this type of infectious material do is that it will stick onto the surface of bacteria, transfer something to it, and then leave it out, right? So with blending, they have ensured that none of the viral particles, right, is going to be still attached with the outer surface of E. coli. Whatever has been gone inside is the only viral component that is being present with the bacterial cell, not the other component which is going to be attached on the surface, right? After the blending, when the viral particles has been separated from bacteria, they have performed centrifugation, right? Now why this centrifugation has been performed? Because we know that viral particles are having lighter density in comparison to the bacteria, which is a bigger cell. So if you perform density gradient centrifugation, which separates the molecule based on the density, so because bacteria is heavy, right, so we are going to have the bacteria settled in the bottom, right? But because viral particles are lighter in comparison to it, would be floating over here in the upper fraction. So if any viral thing has, whichever viral thing, like if it's protein or DNA, whichever is going to show radioactivity in this particular section is going to be the genetic material, right? So when they have, analyzed the particular centrifugation fraction in this particular case where the virus has been labeled with S35, they have found that the radioactivity is being detected in the supernatant. Supernatant means this upper fraction. This is supernatant, and this particular section is pellet. So they have found the radioactivity in the supernatant fraction, and that is how they have ensured that this particular section, this S35 labeled section, hasn't been, gone inside the bacteria. Okay, the blending. Blending is basically an agitation, right? Like for example, these two things are attached together, so I shake them this much vigorously that they'll separate, right? And this blending has been performed in this particular experiment so that the viral particles can be separated from the bacterial particles. It is to ensure that nothing has been attached on the outer surface of bacteria. Whichever has been gone inside is the only viral component that is going to be present in the bacteria, right? Okay. So in the first case, they have found the radioactivity in the supernatant, which is the section of viral particles. So now they are sure of the fact that protein is not the part that goes inside the bacterial cell. On the contrary, when they have analyzed the centrifugal section of this particular set in which the DNA has been labeled, in that particular set they have received the radioactivity in the bacterial cells, that means in the pellet region. So that is how they have given the unequivocal proof that DNA is the genetic material, because this is the material that transfers from one organism to the other, right? In this particular case also, it has been transferred from the virus to the bacteria, right? And this has laid the foundation of identifying that DNA is the genetic material, right? So now we know for a fact that central dogma, the very first molecule that has started it, is the experiment that has been, performed. So now we know everything about DNA, what its structures looks like, why the central dogma starts with it, how we have confirmed that it is the genetic material, right? Centrifugation. Okay, so centrifugation basically is going to separate. Over here we have performed density gradient centrifugation. In simple language, if you want to understand, centrifugation is going to separate the molecules based on their density. So अगर कोई चीज़ बहुत भारी है, it is going to settle down, and something which is lighter is going to float above, right? तो जो चीज़ नीचे settle down हो जाती है, it is similar like sedimentation, right? Something heavier like a stone particle put on in the water, the stone particle is going to settle in the bottom, right? तो similar centrifugation करता है, लेकिन क्योंकि तो हम centrifugation use क्यों कर रहे हैं, simple sedimentation कर दे, क्योंकि ये molecules जो ये particular organism है, they are microscopic, so we need a stronger force in order to sediment them down. That is why we are using centrifugation. तो अगर आप बैक्टीरिया और वायरस को कंपेयर करोगे तो बैक्टीरिया इज अ बिगर स्ट्रक्चर इन कंपैरिजन टू वायरस, राइट? तो बैक्टीरिया कहाँ पे सेटल होगा? नीचे, right? Because it's heavier, and virus being the lighter particle is going to be present in the upper section. Upper section को हम क्या बोलते हैं? Supernatant, and the lower sedimented section is known as pellet, right? So जिसकी भी रेडियो एक्टिविटी, whether it is S35 और P32, जिसकी भी रेडियो एक्टिविटी इस सेक्शन में आएगी तो वो पर्टिकुलर चीज़ जेनेटिक मटेरियल होगी. क्यों? क्योंकि बैक्टीरिया को इस एक्सपेरिमेंट में लेबल नहीं किया है. किसको लेबल किया है? वायरस को लेबल किया है. सो वायरस का ही पार्टिकल बैक्टीरिया को रेडियो एक्टिविटी दे सकता है, राइट? तो पहले पर्टिकुलर केस में क्या हुआ जिसमें S35 लेबल था, उसमें जब इन्फेक्शन किया इसमें और सेंट्रीफ्यूगेशन परफॉर्म किया, सो दे हैव फाउंड द रेडियो एक्टिविटी इन दिस पर्टिकुलर सेक्शन. Supernatant. Supernatant में क्या present है? Viral particles. That simply means that this particular particle was unable to, being inserted into the bacteria. That is why radioactivity यहाँ नहीं यहाँ पे present है. In the second case, when P32 was labeled, in that particular case, they have seen the radioactivity in this section, which simply means कि virus का ये particular particle bacteria के अंदर enter हो गया, and that is how the bacteria gain its radioactivity. So this is how they have proposed the fact that DNA is going to be the genetic material. I hope this is clear to you, Tiasha. All right. So now after this particular experiment has been done, right? So now we are clear with the very first structure with which we have started the central dogma. Now we'll start with the thing that properties of genetic material, right? तो जब भी DNA को genetic material propose कर दिया गया, तो then people have started questioning that why RNA cannot be a genetic material, right? So that is the reason why certain properties of genetic material has been proposed. The very first one is the chemical and structural chemical and structural stability, right? So we are going to compare DNA and RNA over here, that which one of them is going to have all the characteristic. So when we talk about chemical and structural stability, so DNA is more structurally stable in comparison to RNA. Now why? Because firstly, DNA is lacking the oxygen atom at the 2' position, right? That's the first thing. Because of which rest of the OH groups have been indulged into, multiple interactions, like 1' OH is being indulged with interaction with the nitrogenous base. 3'1 is being, engaged in the phosphodiester linkage. 4th one already doesn't have the oxygen. 5th oxygen OH group is being entangled with the interaction with the phosphate group. So all the reactive groups are busy in interaction, and at the 2' position, 2' position, the OH that is going to be present, which could have provided the reactivity, is not being found in DNA. So that's the first reason. Second reason is, in place of uracil, DNA has thymine, which provides extra stability. Then DNA usually is present in the double-stranded form. The hydrogen bonding between the nitrogenous bases is providing the stability. So if I compare DNA and RNA on the basis of chemical and structural stability, DNA is structurally and chemically stable, but RNA is not, right? The second feature that's being there is, the capability to replicate. So replication capability. So both of them can replicate. So this property is being found in both of them. Now thirdly, scope for slow changes, right? Because changes are necessary for evolution, right? So scope for slow changes. So DNA can have slow changes, but RNA mutates really fast because of its self-catalytic nature, right? And other reasons also, but this is one of the reasons. Now fourth thing, should be capable to express themselves, express in the form of Mendelian characters. Right? So we know when we have studied the principles of inheritance chapter. So in that particular point of time, we have studied that he has talked about DNA majority of the time. So this Mendelian character thing is most probably true for DNA part. It's not been infused or not been proposed for RNA specifically, so we cannot give any kind of weightage over here. So अभी अगर हम देखें तो यहाँ पे हम क्या देख रहे हैं कि majority के सारे ही characters DNA जो है वो follow कर रहा है, but it's not being present in RNA. That is the reason why DNA is the genetic material. So they have proposed this particular fact, and this line has been specifically mentioned in NCERT also, that for the storage of information, DNA is the better material, but for the transmission of information, RNA is the better material, right? So this is a really important line, that DNA is a very good material for the storage of information. That is the reason why it's being the genetic material in all the cellular organism. But when we talk about RNA, it is good for transmission of information. That is why you must have seen that when we have written the central dogma, RNA is the middle component which is actually transferring the knowledge from DNA to the protein, right? So then people have came up with an idea of RNA world, right? Because RNA ka structure comparison to DNA is simpler. RNA also has the catalytic ability, self catalysis ka property hota hai uske andar. So that is why it has been proposed ki most probably during the evolutionary scale, initially the genetic material that has came into existence was RNA. Later on, due to stabilization of the environment and all those things, it has been modified to DNA, which is a much stable structure, and that is how the progression in evolution must have been done, right? So this is all about the first molecule with which we have started the central dogma, that's DNA. Now we'll go to the first process of central dogma, and that particular process is replication, right? With which DNA makes its own copies, right? So when we talk about the nature of replication, that how replication is going to be performed, so this particular model was initially being proposed by Watson and Crick when they have proposed their structure of DNA. At that time only they have proposed that what kind of nature the replication is going to show, and that nature is semi-conservative nature, right? Now semi-conservative nature का क्या मतलब है, that if let's suppose I'm going to use this as the parental strands, right? So when the replication is going to occur, which is making the copies of DNA, so over here one parental strand is being used as the template, and against this we'll be having another daughter strand. So in the new copies we'll see one parental copy and one newly formed copy. I am making it with blue not because the sequence has been changed. I am making it with blue in order to show you कि जो नई synthesized copies है, those are going to be like this. ठीक है? Now one important thing again over here is that the replication is going to follow the law of complementarity. Right? This is really important to know that replication follow the law of complementarity, which simply means that A is going to bond with T and C is going to bond with G, right? And vice versa, of course. So इससे क्या advantage मिलेगा? The advantage over here is that if you'll be knowing the sequence of one strand, then you can propose the sequence of the other strand really easily, right? Like for example, if one strand has the sequence as ATGG, so the complementary strand if you have to propose, then it will be TACC, right? So this is really important that replication follows the complementarity, law of complementarity. Now the point is that Watson and Crick have proposed, while proposing their double helical model of DNA, that the replication is going to be semi-conservative, but it was just a proposition. So we needed an experimental proof that yes, the replication is going to be semi-conservative in nature. So for that, the experiment was performed by Messelson and Stahl, right? So in order to perform this experiment, they have chosen the test organism as E. coli, right? Now, rather than choosing the radioactive substances which is going to harm the organism, they have chosen the light and heavy isotope of nitrogen. Now why they have chosen nitrogen? Because we have seen when we are studying the structure of DNA itself, the nitrogen is being found in the nitrogenous bases, a primary constituent of nucleotide, and that's why the primary constituent of DNA also, right? So initially what they have done, they have grown several generations of bacteria into a medium that contains N15, right? And you cannot provide directly the atoms in the culture medium. So that is why the salt, which is having nitrogen's heavy isotope, this is being put into the medium so that whenever bacteria has to synthesize the DNA, it should take up this nitrogen, right? So after several generations, when they have, grown the bacteria into the similar sort of culture medium, then they have found that their DNA is composed of the heavy isotope of nitrogen, right? Now, for the first time, so that they can propose that what type of replication, is being performed by E. coli, what they have done is they have transferred this particular bacteria which is having the heavier density, DNA into a medium that is now having lighter density of nitrogen. Okay? All right. So now if for a time being, let's suppose that कि semi conservative mode नहीं होता, right? तो क्या होना चाहिए? जब अगर ये बैक्टीरिया सेमी कंजर्वेटिवली नहीं कर रहा है रेप्लिकेट, तो क्या होगा? N15 यानी जो पेरेंटल स्ट्रैंड है वो एक साथ रहेंगे, और जो आपके न्यू स्ट्रैंड्स हैं, क्योंकि न्यू वाले क्या कौन सा नाइट्रोजन सोर्स यूज़ करेंगे? N14. सो न्यू वाले एक साथ रहेंगे, राइट, इफ द मॉडल इज नॉट सेमी कंजर्वेटिव. ठीक है, क्योंकि जब ये पर्टिकुलर रेप्लिकेशन हो रही है, वी आर नॉट ऑब्जर्विंग इट थ्रू द माइक्रोस्कोप. ठीक है, हम कैसे आइडेंटिफाई करने वाले हैं कि कैसे रेप्लिकेशन होगा? बाय द हेल्प ऑफ डेंसिटी ग्रेडियंट सेंट्रीफ्यूगेशन, राइट. अगेन वी आर गोइंग टू सी कि जो चीज़ मतलब हेवियर है, दैट इज गोइंग टू सेटल इन द बॉटम, विच इज लाइटर इज गोइंग टू फ्लोट अबव, विच इज गोइंग टू बी इंटरमीडिएट, इट इज गोइंग टू बी इंटरमीडिएट समवेयर, राइट. तो अगर सेमी कंजर्वेटिव मोड ऑफ रेप्लिकेशन नहीं है, तो हमें कितने बैंड्स मिलने चाहिए इनटू द टेस्ट ट्यूब? 2 ऑफ दम, राइट. एक मिलना चाहिए लाइटर डेंसिटी N14, एक मिलना चाहिए हेवियर बैंड, राइट. बट व्हाट दे हैव फाउंड, उनको क्या मिला एग्जैक्टली अपने एक्सपेरिमेंट में? They have found that there is single band of intermediate density. Right? By this they have identified कि यहाँ पे क्या हुआ है? सेमी कंजर्वेटिव मोड हुआ है. सेमी कंजर्वेटिव मोड होगा तो क्या होगा? one parental strand is going to synthesize a new daughter strand. One parental strand is going to make new daughter strand, right? So this is how both of them are having the similar sort of density. तो एक ही band मिलेगा हमें, right? Over here in the density gradient centrifugation, which is not going to be too heavy and not going to be too light, right? In order to make, this experiment more sturdy and more, generalized, they have performed another cycle in N14 medium, N14 containing medium. उसके बाद क्या होगा? Now this is the parental strands, right? Now they behave as parental strand. So if this is a parental strand, new daughter strand will take N14, right? Now if this behave as parental strand, the new daughter strand is going to take N14. Now similarly, if this is going to act as parental strand, the new one is going to take N14. If this is going to behave as parental strand, then again the new one is going to take N14, right? So what we are seeing that we are now having two types of DNA strands, one having N15, N14 having intermediate density, and one N14, N14 having lighter density. And yes, that's what they have found in the centrifugation also. They have found two bands, one with intermediate density and one that is being lighter, right? So this is how they have proposed that the replication occurs in the semi-conservative method, in which every parental strand is being used for the synthesis of new daughter strands. So in the daughter copies, what we are seeing, one parental strand, one newly synthesized strand, right? So this is how they have proposed the semi-conservative mode of replication. Apart from these two scientist experiment, Taylor et al., so they have proposed this particular one on a prokaryotic organism, right? Now, another experiment was done by Taylor et al., right? So this group also performed the experiment, and their test organism was a eukaryotic organism, and that is Vicia faba. And they also came across the same sort of results. So they also say that even the chromosomal replication, the DNA that's been found in the chromosome, also replicated semi-conservative fashion, right? So this is how the mode of replication has been identified. Now, now comes the time to understand that how replication is going to take place, right? So when we talk about the replication, so we know that in order to begin a process, firstly what we need to know that from where we have to start it, right? So if let's suppose we are considering a DNA like this, let's suppose this is, let's say, a linear DNA, or let's try to understand firstly with a circular one. So we know that prokaryotes have circular DNA, right? So in the circle, is there a starting point? So where should we start with replication process, right? So in order to ensure that replication always starts with a particular point, there is a point which is known as ORI. It is known as origin of replication. This is the point from where the replication is going to start, right? And in order to perform this process of replication, the cell should have the abundant supply of deoxyribonucleotides, right? Or we could say deoxyribonucleoside triphosphates, okay? So initially this particular synthesis is being going on, right, in order to create DNTPs, deoxyribonucleoside triphosphates, right? ये पहले बन जाते हैं so that the replication, whenever the replication is going to be performed, this could be used. So these particular molecules has two specific functions. The very first function, that they'll be acting as a substrate, right? Right, like for example, if the sequence let's suppose this particular strand is having the sequence ATGC. Now if I have to, make the copy, so what we need is DTTP. Now this will come and add a T over here. Now next time, because I have a nucleotide against this T, so I'll be needing DATP. This is going to come and join over here. So basically the main polymerizing enzyme used these DNTPs as substrate so that it could be used for the synthesis of new strand. Another function that they serve is acting as an energy source, right? Now energy source means how exactly you're going to add this DTTP over here and then next DATP over here, right? तो क्या होता है, let's suppose कि I have added this T over here. So this is going to have 3 phosphates, right? They are going to bond together because hydrogen bonding is easier to form, right? Whenever there is an electronegative atom and a hydrogen atom, they can form easily. But then how exactly another nucleotide is going to come and join over here? So for that, these 2 phosphate groups are going to be cleaved out. The energy that's going to be released is used for the addition of this A over here, right? Then this A will be having these 3 phosphate groups. These 2 will be broken out to join a C over here, right? So this is how they'll be acting as a substrate also, as an energy source also, right? Now, so again we'll start with this particular molecule, right? This is an ORI. So now we have abundant supply of the DNTPs with us. So we'll start with this particular ORI sequence. So this particular ORI is going to be recognized by a set of molecules, and then from this particular point onwards the helicase will come, right? What is the function of this helicase? The helicase is going to open this up. It is similar like, let's suppose I instruct you to photocopy a page from the biology NCERT book. Let's say I say that you should photocopy page number 72. So firstly what you have to do, you have to open the book, right? And that opening is being done by helicase. So when this opening is going to be done, it will look like this. Sorry. It is going to look like this, right? This is how the opening is going to be done. Now when this opening is done, in these areas there will be severe folds, right? And because this severe folds are going to be there, we need an enzyme that can easily relieve this strain, and that enzyme is topoisomerase, right? It is like something, this screw has been tightened up. In order to loosen it down, topoisomerase can easily loosen it up, right? So topoisomerase is going to lose or relieve this particular strain that is being caused because of the opening of the helix, right? Now this opening is going to create a replication fork. If you will see closely over here, this particular structure is resembled by this, and these 2 are resembling this. This structure is known as replication fork. And within the replication fork, the process of replication is going to take place, right? Now, how this particular process is going to take place? So let's consider this is a replication fork. Now this strand is having a polarity of 3' to 5', and this is having a polarity of 5' to 3'. Now important point over here is that main polymerizing enzyme is DNA polymerase, right? And because this DNA polymerase is going to use DNA as a template. Template means the strand on which it works. That strand is DNA only, so we are going to call this DNA polymerase as DNA dependent DNA polymerase, right? Now this DNA dependent DNA polymerase has certain properties. The very first property is that it cannot synthesize randomly in any direction. It can only synthesize in one direction, and that direction is 5' to 3'. And another thing, it cannot start the synthesis by scratch, it needs firstly a primer, a small nucleotide sequence which could be acting as a cue. Like for example, if you have, let's say, learned a speech or something, and now you're just forgetting the first word, and now if somebody gives you cue, then you'll straight away start with it. So similar way, DNA polymerase cannot start from the scratch. It needs a cue, and that cue is being given by the primer, right? That's a small nucleotide sequence, right? So I'm going to take this as a symbol of primer, right? So now we know that this can synthesize only in one direction, 5' to 3'. Now if you'll consider this particular strand, 3' to 5' one, and you consider this one. Now this over here, the opening is 5'. Against this, the synthesis would be done in 3' direction. अगर यहाँ से synthesis शुरू करते हैं तो इसके एंटी पैरेलल क्या आएगा, 3'. Now अगर हम यहाँ से synthesis शुरू करते हैं तो क्या आएगा, 5', right? So now, because DNA polymerase can synthesize in this direction, so if a primer is put over here, तो DNA polymerase क्या करेगा, synthesis करना शुरू कर देगा, right? But over here, can we put the primer in this particular place? No. अगर हम इस particular place पे लगाते हैं, so this direction is 3', not 5', right? 5' कहाँ पे present होगा, यहाँ पे, right? तो यहाँ पे अगर आप प्राइमर लगाओगे, then this direction will become 5' to 3'. So DNA polymerase can synthesize like this, right? Now, of course, helicase यहाँ पे present है तो वो DNA को further खोलेगा. Now, अगर DNA को हम further खोले, तो now it will look like this, right? इतनी synthesis हम कर चुके हैं. This much we have done, right? This is again 5'. This is 3', right? Now, again DNA polymerase has to synthesize. तो यहाँ पे अभी भी direction क्या बन रही है, 5' to 3'. So the synthesis can continue like this only, क्योंकि already strand है इसके पास. पर अगर हम यहाँ पे देखें, तो अगर इस particular primer को हम use करते हैं, तो synthesis ऐसे करनी पड़ेगी. ये direction क्या है, 3' to 5'. इस direction में तो DNA polymerase synthesize ही नहीं करता. So that is the reason why this particular synthesis is not possible. That means we again have to put a primer over here, and then the synthesis will be done like this, right? Further it will get open, further the same thing is going to happen. So if you could see here, what we are seeing, that in this on this particular strand the synthesis is continuous, right? यहाँ पे क्योंकि एक बार primer लगाया and the synthesis is continuing, so this particular strand is going to be known as leading strand, right? Because over here the synthesis is going to be continuous. On this particular one, क्योंकि आप देखो यहाँ पे breakage में synthesis हो रहा है, this is the lagging strand, right? Because every time whenever the DNA segment gets open up, पहले आपको primer लगाना पड़ेगा and then the synthesis can start. But over here on this particular strand, what's happened? One time you have put the primer and the synthesis is continuing, right? So that is why this is lagging strand. It is lagging because every time it has to start from the scratch. Over here it is being continuous. Now these short fragments that have been made are being known as Okazaki fragments, right? Now we know that with replication हमको क्या करना है, DNA के against DNA के copies बनाने हैं. Now these primers are not made up of deoxyribonucleotide nucleoside triphosphates. On the contrary, they are being made up of RNA, right? Ribonucleoside triphosphates. Or right now I could say ribonucleotides, right? तो ये RNA के molecules हैं, but हमने क्या पढ़ा, replication क्या होती है? The copying of DNA. That means against the DNA we have to make a DNA. That means we need to remove them, right? So then simultaneously when the synthesis is occurring, what happens is that these primers are being removed, right? Now if you remove these primers, right, what you are going to see, the breaks in between the DNA segments, right? That means this particular strand, ये तो चलो continuous बन जाएगा, but what about this particular strand? It is having certain breaks in it. Now any alteration in DNA, we have already studied that in the previous chapter, any alteration in DNA is going to cause mutation, which is going to cause abnormality in the individual, right? That means we have to fill these blanks, right? So further, these blanks are being filled upon by another polymerase enzyme, which is again DNA polymerase. This is DNA polymerase 3 and that's DNA polymerase 1. So this is being, sorry, these gaps are being filled, right, like this, except this particular nick. A small phosphodiester linkage is still being, not get connected, and the reason for that is that when this particular, gap is being filled, what happens is that this DNA strand will be taken up as a primer, and against that the synthesis is going to be occurring. But this is already being formed. So the enzyme cannot make the phosphodiester linkage with it. So every time there will be a small kink which is going to be left out. Then further these small kinks are going to be joined by DNA ligase enzyme, right? And this is how you will fill all the gaps and you will seal these nicks, and at the end of the time what you will be getting, one parental copy and against this a newly formed daughter copy, right? So this is how the complete process of replication takes place, right? And I hope this particular thing is clear. In case you have any doubt, please put up in the chat box so that we could discuss it, right? So this is how we will be, how the body performs DNA replication. One important thing to note over here is that replication occurs for the entire set of DNA, right? It's not like कि heterochromatin में नहीं होगा, euchromatin में नहीं होगा. No, entire DNA is going to be replicated in the S phase, right? So when we talk about the prokaryotic replication, over there we see a single origin of replication, right? Enzyme names. Okay, so when we talk about the enzyme names, the primary enzymes that you should know about is firstly this DNA dependent DNA polymerase, right? This is very important to learn. Apart from that, helicase and topoisomerase are the two things which is important, right? Rest all enzymes, rest details of the enzymes are not being required. Till now it's not been even asked, right? What happens to RNA primers on lagging strand? That's what I've told you, that RNA primers are going to be cleaved out, right? So RNase H enzyme is going to cleave it out, and then that gap is going to be filled. Then there is a nick that is being created, that is going to be sealed by DNA ligase enzyme, right? I hope this is clear to you, Aparna, right? What I've told you, that if let's suppose this is the RNA primer that's been present on the lagging strand, firstly it needs to be dissolved. So an RNase H enzyme, you don't need to learn the name of the enzymes, but you have to that particular thing just removes the primer. Then that particular gap is being filled by another enzyme, right? And then further it is going to be sealed by DNA ligase, right? Isotope experiment once again. Okay. The DNA polymerase 1, 2, 3, it's not being needed because earlier it's been asked, not in the current trends. So बहुत लंबे time से this question wasn't being asked, right? How many times a primer are going to be used in Okazaki fragments? See, for the construction of a single Okazaki fragment, you will be having a primer, right? So number of Okazaki fragments will be dependent upon the type of DNA or the organism or its machinery. We cannot ascertain the number to every organism, but yes, single Okazaki fragment which we have is having the RNA primer along with that particular stretch of DNA that's been synthesized by DNA dependent DNA polymerase, right? Okay. So this is how we will be having the DNA dependent, the process of replication, right? Now we are coming to the next one, that is transcription, right? So when we talk about transcription, that's the second process, right? So till now what we have covered in the central dogma, DNA, and it's been replicated. Now the next process is its conversion into RNA, and that is going to be transcription. This is the second thing that we are going to start now, right? So when we talk about transcription, so this is the process of formation of RNA from DNA, right? Similarly to the process of replication, transcription also follows the law of complementarity, right? This is a similarity of transcription with the replication, right? The differences are, firstly, over here against the DNA, RNA will be synthesized, and replication against the DNA, DNA will be synthesized, right? Why there is a need of many primers on lagging strand? That's what I've explained, like when I'm explaining the process of replication to you, that every time when it opens up, right, then like over here I suppose, every time when the DNA opens up, right, like you could see here, like in this particular case, if you will see, right. So I have already established that this particular enzyme can only synthesize in 5' to 3' direction, right? So in this particular case, the direction is coming out to be 5' to 3', but over here it is coming out to be 3' to 5', right? Against 5' you will be synthesizing 3'. So here you can put one primer, right, and another primer you can put here, so that the direction could be maintained and DNA polymerase can synthesize like this. Now second time when it opens up further, then what will happen? What will happen over here? This much synthesis we have already done, right? Now this is further open up. Again, on this particular strand, the polarity is still 5' to 3'. So DNA polymerase can still synthesize by using this now as a primer. But over here, if this particular primer is being used, then the direction of synthesis would be 3' to 5'. But in this direction, it cannot be used. So that is the reason why another primer needs to be put over here, so that the direction could still be maintained as 5' to 3', right? Okay. So the next one we have is transcription that we were talking about, the making of RNA from DNA, and it still follows the law of complementarity, right? So another difference from transcription, if we compare it with replication, is that in replication, both strands will act as template, right? But in case of transcription, only one strand will act as template. Now why so? Is because if, let's suppose this is one strand, let's say the sequence is like this, right? Let's suppose this is the DNA. So if I'll be considering both of them to be the template strands, so against this, the mRNA that we are going to form will be, or the RNA that we are going to form will be UAG, right? Because now we are making RNA, so in place of thymine, adenine is going to pair up with uracil, right? And again, against this, if I make a RNA, then I'll be making AUC, right? Now, if you consider only these two, can you see the complementarity between them, right? So if both the strand will act as a template, then they'll be going to form two RNAs which are going to be complementary to each other, and they are going to form the double-stranded RNA, which is not suitable for translation, right? So that's the first process. After transcription, we have to perform translation. This particular thing is not suitable. Also, because of the sequence difference, the two strands are going to be complementary to each other, having different sequences. So two different type of proteins will be made, which are going to behave as antagonistic to each other, right? So if certain thing is making protein for synthesis of someone, then the other protein is going to disturb it, right? So overall, there will be no metabolismic process that's going to take place. That is why in transcription, we'll have only one strand that will act as a template, right? Okay, so in order to perform it, firstly, we need to know what is a transcription unit. So when we talk about this transcription unit, we'll be having again two strands of DNA. One is having a polarity of 3' to 5', and another anti-parallel to it, 5' to 3'. Now, whenever we have to, identify a transcription unit or name it, we'll always name it with the help of the coding strand, right? Now, why we are calling it as a coding strand? What is a coding strand? So firstly, when we talk about transcription, so again, it's a polymerization that's going to happen over here. RNA needs to be synthesized. So the polymerization of ribonucleotides needs to be take place. So that is going to be done by DNA dependent RNA polymerase. Right? So this DNA dependent RNA polymerase will start the transcription from the region known as promoter. This is the binding site of RNA polymerase. So similar as you have seen ORI as the starting point of replication, over here the starting point of transcription is going to be the promoter region, right? Now promoter is going to be present at the 5' end of the coding strand, right? And the terminator, which is going to end the process, is present at the 3' end of the coding strand. Now why we are calling it as a coding strand and why we are calling it as a template strand? Template strand, you know, on which we are going to work. So over here against this template strand we are going to make RNA, right? Now why we are calling it as a coding strand or also as sense strand? This is also known as antisense strand. Now why we are calling it like that? Because let's suppose this is the sequence of the template strand, right? So against this the RNA that we can make will be UAGC, right? Now against this, if I have to write the sequence of the strand, another strand, it will be TAGC. Can you see that the sequence of mRNA is same as the sequence of the complementary strand of template strand? Right, except the fact that this is DNA, so it is having thymine, and this is RNA, so it is having uracil. Apart from that, the sequence is exact, right? That is why we call it as a coding strand, because the RNA that we are going to code is going to have the exact similar sequence like the coding strand, except the fact that the RNA molecule is going to have uracil in place of thymine, right? So these kind of questions also come in which either you have been given with the sequence of template strand or the coding strand. If template strand sequence has been given and you have been asked to write the sequence of RNA, right? In that particular case, you have to write the complementary sequence. In case coding strand sequence have been given, then write the exact same sequence. Wherever there is T, replace it with U, right? So this is how the transcription unit looks like. So when we talk about the transcription as such, so prior to that the concept of gene came into picture, right? So what is basically a gene, right? It is to define it in terms of sequence of DNA is really difficult, but generally how we define gene is that the segment of DNA that can, you know, code for an RNA will be considered as gene, right? Whether it's mRNA or rRNA or tRNA, any type of RNA, if it is being coded by a segment of our DNA, then that will be considered as gene, right? Now further, if it is going to be, you know, express itself in the form of protein, then that segment is known as cistron, right? So gene को हम loosely कैसे define करते हैं? A segment of DNA that can code for an RNA, and if it is further going to be code for protein, it will be called as cistron. So एक gene के अंदर cistrons present होते हैं if it is going to be coding for a protein, right? So अगर एक gene जो है वो सिर्फ एक single protein molecule बना रही है, तो that means the gene is going to be monocistronic, क्योंकि उसके अंदर एक ही ऐसा sequence होगा जो कि protein के लिए encode कर रहा है, right? But if it is going to have multiple protein thing, then that will be considered as polycistronic, right? So monocistronic is going to be usually seen in case of eukaryotes, while the polycistronic is usually seen in the case of prokaryotes, right? Eukaryotes and prokaryotes. So this is the basic concept of gene. That is the reason why in the transcriptional unit, this particular section against which we are going to have the synthesis of RNA would be considered as structural gene. Okay, so talking about the UTR, when we are coming to translation, I'm going to tell you why UTR is being present on both sides of the strand, right? Let me first come to that topic, right? Let's first complete this and then we'll come to that topic. All right, so this is being considered as structural gene. Okay, so if we talk about the process of transcription, right? So let's suppose this is the, DNA strand, and this is the promoter region, right? So how an RNA polymerase enzyme is going to know that over here the promoter is present, right? It's a big molecule. It has to firstly identify that the binding site is going to be present over here. So in prokaryotes, what happens is that sigma factor, right, is going to come, identify this particular section of promoter, and this is going to give the signal to the core enzyme of RNA polymerase to come and bind to it, right? Now there is a concept of core enzyme and holo enzyme. Core enzyme simply means that the RNA polymerase is having the units like alpha, beta and omega, right? When it is going to combine with this sigma 1, then it is going to form the holo enzyme, the functional enzyme, right? So when sigma is going to recognize here, the bigger molecule is going to come, right? The RNA polymerase is going to come, bind to this particular site, right? When the binding occurs, then this sigma is going to leave the assembly, right? Now why this is going to leave the assembly? It is similar like, let's suppose you have this particular pocket and something is being present inside like this, and the RNA needs to be synthesized from this particular pocket only. So if this surface is going to be filled by another factor, this particular pocket is not free. That is the reason why sigma factor has to leave, so that this factor can become free, this particular surface can become free for the synthesis of RNA, right? So yes, core plus sigma factor will make the holo enzyme, right? So when RNA polymerase get binded, then what will happen? This is how it will open up the DNA and add the nucleotides further, right? So basically, एक line लिखा है in the NCERT that RNA polymerase is capable of initiating, elongating and terminating the transcription process. So अगर ये line लिखी है, this is okay, but there is a deep understanding in this is कि अगर holo enzyme है तब वो इनिशिएट कर सकता है, कोर एंजाइम सिर्फ इलोंगेट कर सकता है, ठीक है, और टर्मिनेशन करने के लिए वापस से रो फैक्टर्स कम से विद आरएनए पॉलीमरेज कोर एंजाइम, दैट्स हाउ द प्रोसेस विल बी टर्मिनेटेड. तो अगर स्टेटमेंट में लिखा है ओनली कोर एंजाइम इज केपेबल टू इनिशिएट then elongate and terminate the process, then it's not true, right? अगर simply generalized term में लिखा RNA polymerase is capable to do so, then it's completely right? Another thing, over here RNA polymerase is not only polymerizing the RNA like this, right? After initiating वो sigma factor remove हो गया, now it is being synthesizing the RNA. It is not only polymerizing it, it is also opening up the DNA helix, right? जो कि replication में कौन कर रहा था? अलग से एक enzyme था helicase which is performing this function. Over here in transcription, RNA polymerase itself is basically opening the DNA, synthesizing the RNA, and because when it moves ahead, these two strands are complementary to each other, they reseal back, right? So this is how the elongation part is going to take place, and further when the terminator region is going to be present, let's suppose the terminator is present somewhat here, right? Then what will happen? This particular sequence is going to be recognized by the rho factor. So rho factor comes over here and binds to this one, right? So RNA polymerase is moving. It is similar like a truck is moving in a very narrow lane and a big stone is present in the lane because of which the truck cannot move forward. So similar way, this is that big block, this is that narrow lane, the DNA, and this is the truck. When it moves ahead, it cannot pass through it. That is the reason why it leaves the assembly, right? And this particular RNA is also going to be left out, and the process of transcription is going to be completed, right? So over here, RNA polymerase is performing the entire function, right? Over there, multiple enzymes are being involved at multiple points of time, right? So this is how the transcriptional processes take place, right? Now, one important thing over here is that when we talk about prokaryotic transcription, right? So when we talk about the prokaryotic transcription, उसके केस में क्या है कि जो ट्रांसक्रिप्शन है एंड ट्रांसलेशन है, दैट्स गोना अकर इन द सेम कंपार्टमेंट बिकॉज देर इज नो कंपार्टमेंटलाइजेशन, देर इज नो न्यूक्लियस और cytoplasm or any other organelle that's going to be different. Both the processes are going to occur in the same compartment, and that is the reason what happens is that prior transcription is being completed, उससे पहले ही translation start हो जाता है. Another reason why this is happened कि transcription and translation are coupled. Another reason is that when we talk about the RNA of the RNA that's being produced in case of prokaryotic transcription, that particular RNA lacks the intronic regions, right? So another term that we need to know now is exons and introns, right? So exons are the regions in the mRNA that is, or basically exons are basically those regions which can code for a polypeptide, right? Introns are the regions which cannot do so, right? Hairpin loop like formation, ending of transcription, yes. So row dependent ending भी हो सकता है, row dependent termination भी हो सकता है, row independent भी हो सकता है, but because that's not in the purview of NCERT, that is why we are strictly adhering to the termination process that's been mentioned in the NCERT. That's row dependent process in which row factor is going to recognize the termination sequence, and that's how it will block the RNA polymerase further pathway, and that's how the process of transcription is going to be ended, right? It could be done in the same way for the hairpin loop like structure when it is row independent, right? But we don't have to study that particular part here, right? Now coming to exons and introns, exons are those parts which can code for polypeptide, introns are those parts which does not code for polypeptide, right? So obviously if introns are being present, transcription ke baad next process that we have to perform is translation, in which we have to take this RNA as a template strand, and against this we are going to have the formation of a polypeptide. अगर ये sequence express ही नहीं कर सकते अपने आप को in the form of a polypeptide, then why these are being needed, right? So thankfully, or we could say blissfully, prokaryotes doesn't have the intronic regions, right? That is the reason why over there transcription and translation are coupled. So two reasons for it. First, absence of compartmentalization, and second thing is the absence of introns. But in case of eukaryotes, when we see eukaryotic RNA or the primary transcript that is being formed is going to have the introns in it, right? So firstly in that, these introns needs to be removed. Also, because in eukaryotic cell we see the compartmentalization, that means the process of replication and transcription are occurring within the nucleus, while the process of translation has to occur in the cytoplasm. That is the reason why the transcription and translation are not coupled. It's not like that the transcription is still not completed and the translation has just started, right? Okay, so if we talk about the next thing, that is post-transcriptional modification. Now why we are calling it as a post-transcriptional modification? This occurs in case of eukaryotes, right? Because the primary transcript that is being formed in eukaryotes is an hnRNA, not mRNA, right? hnRNA simply means heteronuclear RNA. So heteronuclear RNA mein you will be having both sequences, exons and introns, right? As both type of sequences are being present over here, that's why we are going to call it as an hnRNA. It still has the sequences like these which cannot code for a polypeptide, right? So in the post-transcriptional modification, that means the modification that is being performed after the transcription process is being completed, requires the removal of these introns and joining back of these exons, and this particular process is known as splicing, right? So in splicing there will be removal of introns and joining of exons in a defined manner, right? This is one process. Why it is being required is because these sequences cannot further code for a polypeptide. Now the second post-transcriptional modification is capping, which is the addition of (7'-methyl guanosine cap). this particular unusual nucleotide is being added at the 5' end, right? Why this particular thing needs to be added at the 5' end is because this particular nucleotide is going to be recognized by the ribosomal smaller subunit 18S rRNA that is being present in the smaller subunit of the cytoplasmic ribosome found in the eukaryotic cell. The recognition is going to be done by this. That is the reason why this capping is important. Another reason why this capping is important, in order to prevent the RNA from destruction by the nucleases. And for the similar reasons, another process, that is tailing, is also done, so that it could save the mRNA from the destruction by the nucleases that is being present in the cytoplasm. Reason being, in eukaryotic cell, no nucleic acid floats in the cytoplasm. They are being enclosed in certain organelles only. So that is why nucleases ensure that if any RNA or DNA is coming into the cytoplasm, that is going to be foreign. So they are going to cleave it, right? So it is a defense mechanism so that we could prevent ourselves from infection. But over here what happens is that here both the ends are going to be free, and this needs to be transported into the cytosol so that translation could be done, right? If that is not going to happen, then this particular RNA is also going to be cleaved by the nucleases. So in order to protect both the ends, in one end we'll be having capping, and on the another end we'll be having tailing. Now the tailing is being done at the 3' end, and over here we are going to add adenylate residues, right? So that 200 to 300 nucleotides will add up, so that there will be a long chain which is going to be formed. Why we are not doing that much long thing over here? Because this particular thing is not only preventing the RNA from degradation, it is also acting as a recognition sequence for the ribosomal material also, right? This particular process, why this is important, another reason is this splicing is being catalyzed by ribozyme. Right, ribozyme is when RNA molecule act as catalyst, right? We have studied it while we are studying the RNA world, that RNA can also act as a catalyst. This is one such process in which ribozyme, which is SNRPs, are basically performing the function, right? So this is what post-transcriptional modification, and when this particular one is going to be done, then we'll be getting the mRNA which is ready for the process of translation in eukaryotic cytoplasm, right? So further, the RNA will go for translation, but prior to study translation, we should know the genetic code, right? Now, why we are studying this particular genetic code? Because this is going to tell us that how we are going to decipher the sequence of DNA into the protein, right? So initially, the proposition that what is going to be, like what kind of genetic code we are going to have, is being proposed by Gamow, who is a physicist, and he said that because we have 4 nucleotides and we have got 20 amino acids, so it cannot be a singlet. Singlet means a single nucleotide is not going to code for these 20 amino acids, right? It's impossible. It can't be, a doublet also, because if it's going to be doublet, then 16 amino acids are going to be coded, still 4 are going to be left, right? So it must be a triplet, because only then all these 20 amino acids are going to be coded in the body, right? Because we have all 20 amino acids present in our body. So this was just a proposition, because he doesn't have the experimental proof or anything. So that is why his proposition was not being taken seriously, is because 64 is a very much huge number in comparison to 20, right? So that is the reason why another scientists have been came up, like Nierenberg and Mathai, who have synthesized RNA, poly-U RNA, and told us that how exactly cell-free synthesis can be done of a stretch of a nucleic acid. Then came marvelous work of Hargobind Khurana, who have given the chemical method of synthesizing RNA molecules, right? and he has created the heteropolymer. So he is being credited as being considered as the first person to synthesize an artificial gene, right, or an RNA sequence basically. So with their experiment, it has been identified, and there is one more experiment that has been done by Severo Ochoa, who has discovered the enzyme polynucleotide phosphorylase. This particular enzyme has a speciality that it could synthesize RNA in template independent manner, right? This is really important. Now, when we have seen the process of replication, it was template dependent, DNA was the template. When we talk about replication, transcription, again template dependent, DNA is the template. Only the process of capping, tailing, and if the RNA is being synthesized by this particular one, till now the processes that we have studied, in that I am saying the word only. These three processes like capping, tailing, and if the RNA is going to be synthesized by this enzyme is going to occur in template independent manner, right? So with their experiment, it has been proposed that DNA is going to be triplet, right? So in the simple terms, if I have to understand that how exactly they have deciphered it. So basically, Hargobind Khurana has made this, sequence of a heteropolymer of RNA. So let's suppose, hypothetically, if let's suppose this is coding for leucine and this is coding for serine. Now we know that it's a triplet. But let's suppose for now, let's consider it to be a singlet, right? So pehle unhone kya kiya? He has synthesized it and translated it, and he has found the chain of leucine and serine consecutively, right? Now he said that if it is going to be a singlet, right, then if I remove this particular one, then I should get the same sequence, right? But he has got a different sequence, right? Then if it will be a doublet, then hame same sequence milna chahiye after cutting the thing. He didn't get it. Then when he cut three of them together, then he got the exact same sequence again. That is how it has been deciphered that it is going to be a triplet code. So it's not like it's only the experiment of Hargobind Khurana that has deciphered it, but there's of course multiple contributions has came up with this particular idea that genetic code is a triplet. So that is why we are coming to our next topic, that is the salient features of genetic code, right? So when we talk about is 5' capping and tail is a same as UTR. No, it's not same as going to be UTR. 5' methyl, 7-methyl guanosine cap is a single nucleotide. It can't be a UTR, right? Okay, so coming to the salient features of genetic code, there are certain features. Firstly, it will be a universal code, nearly universal. Why we are calling it as nearly universal? Because we can see certain exceptions in mitochondrial DNA, so universal, nearly universal. Like if I'm talking that it will be triple U, it will be going to code for phenylalanine in all of the organism, right? Second nature, very important, is triplet, जो कि अभी हमने decipher किया that it's going to be triplet in nature. Thirdly, it is going to be punctuation less. Right? That means if I am writing a sequence like this, we are not going to put commas or full stops or anything like that. Also, it is non-overlapping. That means if the reading frame has to be set, it will be read as AGG and GCA, not like AGC, then GCG, then CGC, then GCA, right? So it's non-overlapping. Then fifthly, we'll be having a codon that is AUG. It serves two functions. Firstly, it will be acting as a start codon, and secondly, it will be coding for methionine, right? Then another feature is we'll be having three stop codons, UAG, UAA and UGA. Okay, these are going to be stop codons, right? And these are the ones against which we won't be having any tRNA, right? Another feature, that the code is going to be unambiguous, right? That means if let's suppose U is going to code for phenylalanine, unambiguous or specific, that means triple U is always going to be code for phenylalanine. It's not like that in one organism it is coding for phenylalanine and in some other one it is going to be, you know, coding for alanine. Eighthly, it will be degenerate, right? Degenerate means for a single amino acid you can have multiple codons, like serine has got 6 codons for it, right? The exception over here we could see could be AUG, right? This is the only codon that codes for methionine. So these are the features of genetic code that is going to help us while we are studying the process of transcription, right? Amber, ochre, opal, we are not using these terms anymore, so it's not in the purview of this, right? So we don't use them, we are simply going to use these codons, and these are the 4 codons that are paramount to learn. You should know them, right? So those 64 is now setting up the criteria that apart from these 3, right, rest 61 are the codons against which there will be an amino acid that is going to be coded up, right? Now coming to, as we have learned the genetic code features, this is the table that has been deciphered not by the experiment of few scientists but the work of a series of scientists who have came up, and finally the congregation or the amalgamation of all that data have created this particular genetic code table, right? In this again, as you could see, AUG, UA, UAG and UGA, these are the ones that you need to learn, because every time whenever translation starts, it will start with this and it will be end up on any of these, right? Okay, now coming to the structure of tRNA. Now why we are studying it? So firstly, when we talk about translation, all 3 types of RNAs, whether it's tRNA or it is mRNA or it is rRNA, all of them are going to be used in this particular process, right? Over here in the process of translation, mRNA will act as a template molecule. rRNA will help in identifying the starting point and then further, facilitate the process. And this tRNA will act as an adapter molecule, right? So if you see the structure of tRNA, again the presence of this particular structure was proposed by, Francis Crick. So this particular molecule helps in taking up the amino acid and helps to, convert that RNA sequence into the protein sequence, right? So if you see the structure, this is a 2D structure. If you will see, it will look like clover leaf, right? This is a 2D structure. If we talk about its 3D structure, it will look like inverted L, right? Inverted L shaped. So if you see this particular structure, the very first loop that you see over here from the 5' end, it's a D-arm loop or DHU loop, dihydroxyuridine loop. This particular loop helps in binding with amino acyl tRNA synthetase enzyme. Why this enzyme is necessary? It will going to add up or going to charge this tRNA. Charge this tRNA means going to add this particular tRNA with an amino acid, right? Then coming this loop, anticodon loop. This is really important because this loop helps in binding this tRNA with mRNA, right? Whatever sequence is going to be present on the mRNA as a codon, its exact complementary sequence is going to be present in the anticodon loop. Then this is a variable arm, right? Then coming to this loop, T-Cy-C loop, this helps in facilitating the binding of tRNA with ribosome. So this is a ribosomal binding loop, right? Then this 3' arm, this is known as acceptor arm, basically amino acid acceptor arm, where you are going to have the addition of amino acid, and this enzyme will help in doing that, right? So now we know the structure of tRNA, We have already created mRNA. rRNA is already being present in the ribosomes, which you must have read in the cell chapter, are the protein synthesizing factories. So all 3 RNAs are at one place. Now we can begin with the process of translation, right? Okay. So when we talk about the process of translation, so this is, let's suppose, the mRNA sequence. This is 5 prime, this is 3 prime, right? Why this is 5 prime to 3 prime? Because like DNA dependent DNA polymerase, DNA dependent RNA polymerase can also synthesize in this direction only, 5 prime to 3 prime, right? So firstly, what will happen? The starting sequence needs to be recognized, right? So in case of prokaryotic translation, that sequence will be SD sequence, that is going to be recognized by the smaller subunit of the ribosome. But prior to this particular process begin, right, prior to translation begin, there is certain preparation of translation also going on, and that thing is known as first phase of translation. Before the recognition happens or anything happens, right, the certain preparatory part is known as first phase of translation. In this first phase of translation, the amino acylation of tRNA takes place, right? Amino acylation of tRNA simply means when the tRNA is being attached with the amino acid, its cognate amino acid, right? That is known as amino acylation of tRNA or charging of tRNA. This needs to be done prior to the start of translation, because this charged tRNA is the molecule that can convene the process of translation. If you will be seeing the uncharged form of it, it is incapable to convene the process, right? So firstly, in the first phase of translation, this charging of tRNA will take place. Then only the translation can begin, right? So again, let's make an RNA over here, 5 prime to 3 prime. So firstly, as we have seen the process of replication and transcription, there has to be a starting point, right? And over here, the starting point is present at the 5 prime end, which is going to be recognized by the smaller subunit of the ribosome, right? Now, translation, similar like replication and transcription, is also an energy requiring process, right? Over there, you must have seen that in case of replication, the energy is being provided by the deoxyribonucleoside triphosphate. In case of transcription, that energy is going to be provided by the ribonucleoside triphosphates. Over here, ATP is directly going to be utilized. Firstly, it is being utilized when we were performing the first phase of translation. It's an energy requiring step. Then further, when the smaller subunit comes into contact with the mRNA, as soon as this happens, the process of translation begins, right? This is the primary step of the initiation process, right? This line has been explicitly mentioned and has been came in the PYQs as well, where they have written that as soon as the ribosome, the smaller subunit of ribosome encounters the mRNA, the process of translation begins, right? So when it is being recognized, right? So these recognition sequences, right, or you could say the sequences like only ribosome is not the thing that is going to facilitate the process of translation. Translation factors are also there. So the recognition sequences of those, their binding sites, all of them are going to be present in the UTR regions, right? So UTRs are being important because these are providing the additional sites so that the translation could be an efficient process. So the binding sites of many molecules, specificity driven things, all these things are being dictated by UTRs. So UTRs, being untranslated regions, are not going to contribute directly to the formation of polypeptide, but are going to provide the regulatory sequences, or you could say certain recognition or specific sequences which are being utilized during this particular process. Because we are not studying the translation in detail, because that's a really broad topic, Also, it's not been in the purview of NCERT, so that is why we are not going into the details. But as someone has asked earlier why UTRs are so important, is because of this particular reason, and that's the reason it's been present on both the sides, because at certain point of time the translation process has to end also, right? It's not like it has begun, so it will be, you know, going till here also, like the entire, you know, tailing thing also needs to be translated. So that's why UTRs are going to block it up and going to give the efficiency to the process of translation. So as soon as this particular thing has been binded up, the smaller subunit has been binded up, so the rRNAs that are being present within the ribosomes helps, you know, helps in the binding of the smaller subunit of ribosome to the mRNA molecule. Now when this is being done, then first tRNA, that is the initiator tRNA. Initiator tRNA means which is going to have methionine at its 3' end and also going to have this as an anticodon, right? This particular tRNA is going to come and bind to the first one over here, AUG, right? That's the start codon, right? So this assembly will move here and the tRNA will also bind here. This binding, this formation of this particular complex where the 30S, you know, or 40S in case of eukaryotes is been binded with the mRNA along with this tRNA. This particular complex formation is also energy dependent, right? So what will happen after this complex is being formed? Only then the larger subunit will come, right? So this complex will be formed in this way. Really sorry, I have made it a bit incorrectly. After the recognition, this sequence is going to, this particular ribosomal subunit is going to encounter this AUG, the start codon, right? This will move, and this is when the tRNA, the starting initiator tRNA will come and bind to it. After this, the larger subunit also comes and binds to this. This completes the, you know, formation of the translation assembly, right? This is the translational assembly. Now this subunit will move further. So this translocation is also important, and this translocation is also an energy dependent process. Now further when this moves, right, let's suppose this is AUG. Now let's say over here it is CUC. Randomly I'm taking. So now this assembly has moved over here. So within the larger subunit there are certain pockets present like this, right, sites. So the site in which new tRNA comes in will be considered as A site, also known as acceptor site, because it's going to accept the new tRNAs as soon as they come into the assembly. The site prior to that is a P site. P site is the site where the initiator tRNA also came in. Why it is known as P site? Because over here peptide bond formation is going to take place and peptidal transferase is going to perform its function, right? So we know that at this P site already an RNA is present, which is methionine, right? Let's make it on another screen to understand it well. So this is AUG, this is CUC. Let's suppose randomly I'm writing certain sequence GUC, CAG, UAG, right? Like this. So right now our assembly looks like this. This is the P site, this is the A site. Here the tRNA already being present with methionine, you know, attached to it, because it is having anticodon with it, so it will be binded like this. Now a new tRNA will come over here. I don't know what does it code for, but let's suppose it is coding for serine, right, hypothetically. So now what will happen? The peptidal transferase enzyme, in case of prokaryotes, this function is being facilitated by 23S rRNA, which is present in the bigger subunit, larger subunit, right? So this is another example of the ribozyme that will help in catalyzing a process. So over here, what peptidal transferase will do? It will form a peptide linkage between these two amino acids, right? When the peptide linkage is being formed entirely, then what will happen? There will be a cleavage reaction which will cleave this amino acid from this tRNA, right? So what will happen now? this tRNA is now getting cleaved from it after the peptidal bond formation. Now if you will see this tRNA, is it charged or uncharged? Now it has become uncharged. And when we have started this process, I've told you that whenever the tRNA is present in the uncharged process, uncharged form, not process, then it won't be able to convene the process, right? So now this will leave the assembly and this translational assembly will move forward. So now this translational assembly will reach over here, CUC, AUG, GUC, right? This is the smaller subunit. This is the larger one. This is the uncharged tRNA. Now this has been came to the P site. This tRNA has got serine and methionine attached to it. And this is now come at the A site, the new codon. So the new tRNA will come, bind over here. Let's suppose it is coding for isoleucine, hypothetically again. So at the A site, a new tRNA will come, and from this side, the uncharged tRNA will go away, exit the translational assembly. So now again peptidal transferase will form the bond between these two amino acids, and after this bond formation occurs, there will be a cleavage that is going to take place, and this is how the polypeptide chain is going to grow, right? Now let's suppose when all this happened, right? So let's see what sequence we have written. Okay, so let's suppose AUG, maybe I'm writing certain other sequence, but let's say, right, till now the assembly is here, right? 3 codons could be taken at once in at one time, right? So this, let's suppose, already has isoleucine, serine and methionine. From here the uncharged tRNA has already been moved out. Now if you will see that at the A site, now you are having a stop codon, right? Till the stop codon is not going to be encountered at the A site, the process will continue. Every time a new tRNA will come, peptidal transferase will made a peptide bond between the 2 amino acids, right? The one at the P site and the one at the A site. The time it will be formed, the tRNA that is present at the A site will show a cleavage reaction in which the amino acid got detached from it. It will become uncharged, and further when the assembly moves, that uncharged tRNA will move away. Now this is the first time when a stop codon is being encountered by the A site. At this particular moment of time, because we know that stop codons don't have any tRNA for them, so here release factor will come and bind, right? Now we know that release factor is not tRNA. So now peptidal transferase cannot perform its function, right? So that particular process is not being stopped. Now when that particular process is being stopped and no tRNA is coming over here, the cleavage reaction still take place after a while, right? So what will happen? This particular chain is now free because it is now having no attachment, right? Then, sorry. Now if you will talk about this particular tRNA, it has been uncharged. It is also going to leave the assembly. Now this particular ribosomal subunit, whether it has any type of tRNA or any kind of molecule, no. So this is also going to disassemble. So this is how the process of termination of translation also takes place, right? So we have created the protein or polypeptide chain that is being required for the functioning in the body, right? I have created for 3 amino acid long. This is not even acceptable. It's a very long chain, but because we are trying to understand the process that how it is being done, that is the reason why I have made it for 3 amino acid long, right? So this is how we accomplish the process of translation. After this polypeptide chain is being made, then it will undergo further modifications or folding and other type of modifications, so that it will become the exact functional protein molecule which is being required, right? Like for example, if it has to be made like hemoglobin, right? So primary folding, secondary folding, tertiary folding, quaternary folding, all these things needs to be done, but it will be done only after the translation process is being completed. And the same mRNA can be used for the translation multiple times, right? It's not like that one time it is being done, so you need a new mRNA, right? The same mRNA could be used multiple times. After the usage is being complete, it is being cleaved out, and then the nucleotides will again be restored back and could be used for other processes again, right? So this is how the translation will occur, and this is what completes our central dogma, right? Now the question appears that whether this transcription translation is going to occur all the time. So that's not the thing. And how exactly it has to be ensured that this time the product is being needed, so this time it has to be produced, this time the product is not needed, so we need to stop the thing, right? So all these things will come under the regulation of gene expression, right? So next thing that we are going to talk about is the regulation of gene expression. Sorry. So when we talk about regulation of gene expression, it occurs at multiple points, like during the process of transcription, right? So we could stop the process of transcription if the protein is not being needed. Then it doesn't needs to be transcribed. Why to waste energy in that? Then we could, control it at post-transcriptional modification level. We could regulate the process while the mRNA is being transported, right, from the nucleus to the cytoplasm, right, from nucleus to cytoplasm. Then we could regulate it, or we could regulate it at the translational level too. So in the eukaryotic system, all of them are the primary points where you can control the process. At different set of conditions, we will control the process at different times, right? In prokaryotes, the predominant site where we can control it is the transcriptional initiation, right? This has been mentioned in NCERT also, that the predominant site of gene regulation in case of prokaryotes will be the transcriptional initiation. That's where you can control the entire process. If it's needed, perform the process. If it's not needed, then don't perform it, right? Then the next thing, as we have already studied about the central dogma, and we are talking about now the gene regulation, a very important thing that came into our mind is the Operon concept, right? So this is, what is Operon basically? It is a set of genes that are regulated transcriptionally. So it's a transcriptionally regulated system. And the very first example of it is being discovered by Jacob and Monod. They are the ones who has discovered the first transcriptionally regulated system, which is basically Lac Operon, right? So prior understanding this, we should know that what all we consider, what set of genes we are considering during the Operon thing, okay? So that is going to be, first gene type would be regulator gene. This is going to produce a regulator molecule which is going to decide that whether the structural genes needs to be transcribed or not, right? So the regulatory molecule is going to be synthesized by this particular gene. Then we have operator gene, right? This is the gene that is going to interact with the product of the regulator gene, so that it could control that whether it is going to be transcribe the structural genes or not. Then we have our promoter gene, which is going to bind with the RNA polymerase. It will provide the site for the binding of RNA polymerase. Then we have structural genes, which actually produces the proteins that are being required to control the metabolism of certain metabolite. Then we have, of course, terminator gene, which is going to end up the process of transcription, right? So terminator is not that much of importance in the operon concept, but these are, right? So the operon that we are going to study now would be Lac Operon. So Lac Operon talks about the metabolism of lactose in the cell, right? So it is going to talk about the lactose metabolism. So it is very simple that if the lactose is present in the system, only then it needs to be digested. If it is not present in the system, it doesn't need to be digested, right? It's a very simple logic, but that is what Lac Operon controls, right? So if, let's suppose, firstly we are taking the condition when lactose is not present in the medium. Right? So if lactose is not present in the medium, this is how a Lac Operon looks like. So let's suppose this is terminator. So this is A, Y, Z, operator, promoter, operator, right? So this is how the gene setup will look like. So, sorry, this is promoter, not operator. Really sorry about that. So, firstly, we have these three genes, Z, Y, A, right? These three are important for synthesizing the polypeptides that are being required for the metabolism of lactose, right? And as you can see over here, that Z, Y, and A, all three of them are under the control of a single promoter, right? Which simply implies the fact that it is a polycistronic gene, because when we have seen the transcriptional unit with a structural gene, how many promoters we have? Single. So if there is a single promoter, that means it will be a single gene, because gene is the stretch of DNA that's going to code for RNA. So for all three of them, there will be a single RNA that's going to be produced, mRNA going to be produced. And then further they can make multiple proteins out of it. So this gene is basically polycistronic in nature, right? So ZYA are the structural genes. Upstream to that, anything that's being present on the left side of the genes that you are considering would be considered as upstream, and whatever is going to present on the right hand side, it's like a number line. So whichever gene is like under study or main important genes, those will be considered as a 0 on the number line. Whatever is present on the left hand side would be considered as upstream, and their location is always marked by a minus sign. Like let's suppose if I say that promoter is present 35 bases upstream, so I'll be writing it as like this, that promoter is present at minus 35. So count 35 nucleotides like this, and after 35 it will be present. And for the downstream we use the symbol plus, right? So upstream to that we'll be having an operator gene, right? So over here we are loosely defining the term gene, right? It's basically a region. It's not going to code for an RNA. We are loosely defining this term gene, they're not the exact one. So this is operator region, then this is a promoter region which is going to give the site for the binding of RNA polymerase. Now upstream to this entire section we have another gene which is a regulator gene, right? And of course it is under the control of a promoter only, right? So firstly, right, because of the upstream placement and for the greater effectivity, firstly what happens is that RNA polymerase, which is specific, like the sigma factor is going to recognize it, and the RNA polymerase will bind to it and going to transcribe this section prior to this section, right? So when the transcription takes place, it is going to produce an mRNA. Further, when translation occurs, it is going to create a molecule, that is the repressor, right? Now this repressor go and binds to this operator region, right? Now the RNA polymerase that is going to be specific for this one, right, that's going to come, and because these promoter and operator regions are almost overlapping, RNA polymerase does not get the perfect site to bind. It's like you have to sit on a chair and half of the chair is being, filled with bag or any other thing. So you don't have the ample space to sit upon it. So similar way, because half of the site is of course being, clogged by this repressor molecule, so RNA polymerase is unable to bind to this promoter site. And because RNA polymerase is not binding to this site, of course, transcription of these genes are not possible, right? So that is why transcription is not going to take place here, right? So if I consider, let's suppose 100 times this operon has to work out, let's suppose. So 99% of the times this repressor wins the race, goes and bind to the operator and won't allow the RNA polymerase to bind to it. But 1% chance are going to be the case where repressor is unable to bind, so then RNA polymerase will bind here and can transcribe it. ठीक है, but वो levels बहुत कम होते हैं, very small levels of the products of these particular molecules. I'll tell you that why that happens. ठीक है, but now comes the point. अभी तो lactose present नहीं था system में, that is the reason why these products of these particular genes are not required. But now if I consider the second case, when lactose is present in the medium, then what will happen? Oops, sorry. Then what will happen? So sorry, I think it's not been. So if your lactose is being present in the medium, then again what happened? Transcription occurs, translation occurs, but this time the lactose, or specifically we could call it as an allolactose, this is being present in the system, then it won't allow it to bind over here. Why? Because firstly it will go and bind to the repressor molecule. So let's suppose repressor molecule is like this kind of a molecule which goes and bind to the DNA molecule like this. Now if this particular site is being clogged with the inducer molecule, it won't be able to bind to the operator region. So this operator region is now free. Now if this is free, so RNA polymerase can easily bind and can facilitate the process or catalyze the process of transcription of these genes. Now further, when translation occurs, then this Z section is going to code for beta galactosidase. This enzyme is important because it will break lactose, which is a disaccharide, into its monomeric unit glucose and galactose. Then this Y section codes for permease enzyme. This permease enzyme is responsible for increasing the permeability of the cell towards these beta galactosidase sites, not sidase, sites. Like if lactose wants to enter into the cell, it's not a smaller molecule, so it needs the facilitation of protein to get enter into the cell, and that's what permease performs. It will allow the entry of lactose into the cell. That is the reason why that 1% of the chance, even if lactose is not present in the medium, the genes synthesize this permease enzyme so that whenever lactose is going to be present in the medium, it could come inside, right? Then more and more permease will be formed and more lactose could come and could be metabolized inside the cell, right? So this permease is going to increase that, and this then A gene is going to code for transacetylase. Right? So this is also important. So this is how the lactose metabolism is being regulated. So whenever the lactose is not being present in the system, in that particular case, the regulator molecule is not allowing these genes to transcribe by, binding its product with the operator gene, which will block the site for the binding of RNA polymerase. But when lactose is present in the system, in that particular case, what happens is that lactose doesn't allow the repressor, it will conformationally change it so that it won't be able to bind to the operator region, and that's when RNA polymerase can bind to it and can cause the transcription, and further the translation also going to be take place, right? So this is what Lac Operon is all about, right? Now coming to another important topic, that is Human Genome Project. Now this particular project has got its relevance because it is providing us the insights about that what all is being found in the genomes of human. And this is being important because of course we know that genome or gene, or what we could say the genetic material, is the console substance which is going to control all the activities of our. Know it in detail, we'll be more capable to understand that how exactly all the functioning in the body is taking place, right? So that is the reason why this project has been proposed, right, and has begun in 1990s. So the primary objective of this particular human genome project was firstly to identify all the genes, right, that what all genes are being present. Identifying all the genes that are being present in the human system, and then to create databases for it, right? Its entire analysis should also be there, so analysis tools needs to be developed, right? Then ethical, legal and social issues needs to be addressed, right? Now why all this is being happening is because when we talk about human genome, so we know 6.6 into 10 to the power 9 base pairs in the diploid set, and it's been present in a single one. We have a whole lot of DNA present in our body, so the data is really huge, right? So that is the reason why a database needs to be created. It cannot be done in books, right, or in pages. So proper tools, analysis tools, or the creation tools, all those things needs to be made during this particular project only, because prior to it none of the infrastructure was there, right? At the same time, because it is involving humans, so the ethical issues, the legal issues, the social issues, all these things needs to be addressed. Why? Because this particular project is not going to be conducted by a single country or a single lab or a single person. It needs a whole lot of manpower so that the accurate analysis could be done. So right, so for that particular amount of man-force, different countries needs to be, participate in this one. So every country has different set of rules. So those, issues needs to be addressed. So with the primary objective of identifying all the genes and to annotate them, that what all functions it perform, this particular project has been launched. Now with this particular project, after this, certain other non-human model organisms are also being sequenced using the same sort of techniques that is being utilized during the human genome project. So if we talk about the methods that they have took, so initially they have took the idea of ESTs. ESTs means express sequence tags. That means all the genes that are expressed as RNA, right? So this particular one that all the genes that are expressed as RNA is being taken as the initial approach, right? So what is the approach? The basic biotechnological approach. I think you must have studied in the biotechnology that firstly we will take the desired gene, then we put it into the vector, then we take a particular host, then transform the host with that recombinant vector, and then try to see the expression. Then if the expression takes place, then we'll make the libraries, or we can have the upscale production of that particular thing. Similar sort of methodologies, but over here the desired genes are only the genes that can express themselves as RNA, right? But if you could have seen, अभी तक हमने जो process पढ़ी, we have seen this particular thing that जब हमारा initial DNA sequence है, let's suppose this one. So now the transcription that is being taken place, that's hnRNA, let's say, right? It has intronic sequences as well. So, but in the cytoplasm from where we could extract the RNA, right? Why the mRNA can be extracted easily as mRNA? Why it could be easily extracted in the form of mRNA is because mRNA is having At 5' end it is having the cap, and at the 3' end it is having a huge tail, right, poly A tail. So that tail can easily be extracted out, right. It is a very easy symbol to extract that out. So that is why they have said ki as whatever genetic content is going to be present in the humans is going to express itself in the form of RNA. So let's isolate that mRNA, and that's how we are able to identify the entire genome. But they have lacked this particular information that the hnRNA that's being synthesized from the original DNA has got intronic sequences as well. So they are going to be cleaved. Also, the entire stretch of DNA is not transcriptionally active, right? You have seen the heterochromatin regions as well. That is transcriptionally inactive. There are certain other regulatory regions also which are transcriptionally inactive. Matlab, they are not going to be directly forming the RNA. So, if you are going to consider only these sequences, the entire and exact sequences are not going to be explained, right? So, that is the reason why later they have changed the approach to sequence annotation, which is the blind approach to, you know, identify and decipher and sequence the entire sequence, and then assigning the functions to each one of them, right? Like for example, let's suppose this is the long stretch, and this particular sequence, let's say, code for P1 protein, this doesn't code, this does code, this doesn't code, things like this. So sequence annotation was the approach in which they were trying to identify the entire stretch, entire sequence of the human genome, right? So this was the methodology. Now for this methodology, as I have told you that this is the general biotechnological approach in which firstly we'll have to talk about the desired genes. So initially the desired genes are the ones that can code for RNA. Later it will be like आपने एक cell में से DNA isolate किया, cut into several pieces, and all of them will now be considered as the gene of interest. Now it has to be, you know, put it into the vectors. So the vectors that have been taken into consideration during HGP were YAC, BAC, and later MAC also. Now what is YAC? Is yeast artificial chromosome or bacterial artificial chromosome, right? These has been taken up, reason being आप normal plasmid नहीं ले सकते जो आप biotechnological processes में लेते हो, like you must have seen PBR 322 and so on. You have to take these vectors because firstly these vectors can carry bigger gene sizes which cannot be carried by smaller plasmid like PBR 322. So these have been specifically constructed during the time of HGP, and here we are calling it as yeast artificial chromosome is because the ORI sequence that's been present in this particular, you know, vector is taken up from yeast, and over here it is being taken up from bacteria. Later on, when this particular, you know, project has been progressed, MAC has also been made, mammalian artificial chromosome, by taking the ORI sequence from that and then construct that particular sequence so that the bigger gene sizes could be transferred to the host. The hosts that have been taken up are bacteria, E. coli, and basically and yeast, right? These were the host organisms in which the expressions has been tested, and then finally the sequencing has been done according to the method that has been proposed by Frederick Sanger, right? Later on, automated sequencers have already also been made, and the entire, you know, genome has been sequenced up, right? So this was the you know, setup of the this has been the setup of the human genome project. This all methodology has been, you know, performed so that they can identify that what all is being present in our genome. And the last chromosome that has been, you know, sequenced was chromosome number 1, whose sequencing is being completed in 2006. But the project submission report was already been submitted in 2003. But the project is still going on, right? And many researchers are still going on, right? But according to the submission report that has been presented in 2003, they have proposed certain, you know, features of this particular one, certain findings of this particular project, and those are so the findings or the salient features, salient features of Human Genome Project was that firstly the genome is containing somehow 3147 something 0.9 million base pairs. Maybe I'm forgetting the data, 74 or 47 sort of million base pairs are being present, right? Then important finding is that an average gene is consisting of 3000 base pairs, which earlier used to be estimated to be bigger size. Then third finding was that 30,000 genes approximately have been found out, right? Which were initially estimated to be 80,000 or 1 lakh or so on, right? And the longest gene that has been identified was the gene that codes for dystrophin, right? This is the longest gene that they have found out. Another finding was that the maximum number of genes are present on chromosome 1 and the least are present on Y chromosome, right? Then they have identified that there are still genes whose function needs to be discovered, right? And less than 2% genes basically code for proteins, right? A major part of human genome is made up of repetitive DNA, right? This is the major chunk. Initially, it's been considered as junk DNA, but still the findings on the experiments are going on to identify that what all it could offer or, is important for the survival or the existence of the human species. So these were the findings of the HGP. Also, one more finding that has been proposed was the presence of SNPs. At 1.4 million locations, the SNPs were found, right? So these were the findings that has been proposed when the submission report was being submitted, and it has given a very idea that the human genome is vast, really vast, and all the aspects cannot be addressed at the same time. But the good thing about Human Genome Project is that it has opened the aspects for multiple researchers specifically related to disorders and how exactly we could contain or evolve or can better the species, right? And other organisms are also being, the genome of other organisms can also be sequenced, because this has created the infrastructure for sequencing of all of them, right? So this has widened our aspect and actually created a vast horizon for the theranostics things to be take place. So this is a very important project with respect to the betterment of the human species as well. So now after the Human Genome Project is being done, this is being done, and because of this all the knowledge, the transcription, translation and all these things have got more importance, right? And we have understood it in more detail. Now coming to this, one important thing that has been came as a important proposition of this particular HGP was the repetitive DNA, right? So this less than 2% which codes for proteins is of course important for our body and for every other organism. What exact advantage does this repetitive DNA can give us? So we know that all the humans are having approximately similar type of genome set, but still we are looking different from each other, right? That's what the foundation of variation is. We have studied an entire chapter of it, principles of inheritance and variation. So still that variation exists, right? So where does that variation comes from? Maybe from this repetitive DNA, right? So that is why this repetitive DNA is being utilized for the process of, or I should say, this is being utilized for performing the process of DNA fingerprinting, right? So the next topic that we are going to take up would be DNA fingerprinting, but prior to that we should know what's the satellite DNA. So if you will actually segregate this, right, so from the bulk DNA you will be finding a smaller peak of it, and that is what a satellite DNA is all about. So satellite DNA, if you will see, let's suppose if this is the chromosome, these are the telomeric regions which can contain satellite DNA. Centromeric region, the heterochromatin region also contains it, right? So these particular stretches which I am showing here are also showing the repetitive DNA, right? And this is how it will look like, right? So if you could see this repetitive DNA, you can clearly see the sequence is AATCCC. Again, it has been repeated AATCCC, and it could repeat multiple number of times, right? So this particular repeat, right? This one, this is known as the tandem repeat. So over here the tandem repeat is 2 because this sequence has came twice. If it could have come more than that, then it could be 4 or 6 or maybe that. That is going to define the tandem repeats, right? So when we talk about the satellite DNA, the types of it, we can have 2 types of satellite DNA based on this base composition, length and number of repetitive units. It could be mini satellites or VNTR, or it could be microsatellites or STRs, right? So mini satellites, the repeats would be 10 to 100 base pairs, and the size would be approximately 0.1 to 20 kb. And if we talk about STRs, the length will be 1 to 6 base pairs, and it will be less than 1 kb, right? Now with this, we are not able to understand that what VNTR and STR is all about. So let's see what exactly does it look like. So when we talk about mini satellites, so let's suppose this is the one sequence, right? This is the repeat size, so 10 to 100 base pair. Let's suppose this is made up of 100 base pairs. So now it is repeating 1, 2, 3, 4 times. So VNTR, it has been repeated 4 times. So the VNTR is now becoming 4, right? Now talking about the STR, now if it is a small sequence, like agar yahan pe dekhe, the repeat size is of almost 6 base pairs. Over here it is a 1 base pair. So this is repeated 8 times, this is repeated 20 times. So for this, SSR or STR becomes 8, and for that the SSR or STR becomes 20, right? So this particular VNTR is being used for DNA fingerprinting. So basically the basic principle of DNA fingerprinting is DNA polymorphism. This was the principle for Human Genome Project also, right? The DNA sequencing under Human Genome Project also. So DNA polymorphism means the slight differences that arises due to mutations that occurs in all of us, right? And because this polymorphism is inheritable also, so that is going to give us a wider, idea that how exactly the genes are being transferred, and we are also able to identify that whether 2 organisms belong together from the same family line or not, right? We are able to identify that whether a particular organism is related to this one or not. We are also going to identify, that if different DNA samples has been given, so which DNA sample belongs to whom, right? So DNA fingerprinting has got wider application, and DNA polymorphism forms the basis of it, right? Now, one more thing is that when DNA polymorphism is a mutation that occurs at a higher frequency in the population, right? Because if it is lower in the population, it won't be traceable. So DNA polymorphism is being considered only if these particular mutations or the variations are present in the higher amount in the population, more than 0.01%, right? So when we talk about this DNA fingerprinting, so this technique was this DNA fingerprinting technique uses VNTR, right, as the identification identification sequence, which will help to identify that whether the DNA that has been isolated is belonging to the organism or not, right? So let's start with the process. If we talk about DNA fingerprinting process, then firstly, let's suppose we have found a DNA sample on a crime site. Let's suppose there is a blood stain or maybe hairs or something like this. We have found from the crime site. Now we will isolate the DNA from it, right? The very first step would be isolation of DNA. right? Now after we isolate it, the second thing that we do is we'll be going to cleave it with the help of restriction enzymes. Right? Now what does it is going to provide us? Because DNA is too huge, right, and when we'll analyze it with the help of, let's suppose, electrophoresis or something like that, then this much bigger DNA molecule cannot be separated out properly. That is the reason why, in order to study it, like for example, if I say that you have to study science, so science is too huge. That is why you have bifurcated into certain streams so that you could specialize yourself into one of them, or maybe, you know, read them accurately, right? Even your biology book has been divided into certain chapters so that you could study the topic in detail, because it is bifurcated. It is much easier to study the things when it is bifurcated compared to when it is being, you know, presented as a bulk. So that is why, because the DNA is going to be a bulk molecule, in order to understand, in order to analyze it, it is better to cleave it into smaller pieces, and that could be done with the help of restriction enzymes. Now after this has been done, they are going to be separated by using the process of electrophoresis. Right? Now electrophoresis basically separate the molecules on the basis of charges to mass ratio. So because DNA molecules are having the negative charge, so their separation is based upon the mass only, and so the bigger molecules of DNA, let's suppose if this is the gel, let's suppose, so the bigger molecules are going to stay like closer to the wells where the samples have been loaded, and the lighter ones are going to be far away from the wells because they can move easily, right? So we'll do the separation by electrophoresis. So now why this particular DNA has been taken up? So this DNA which has been taken up from the crime site is being used. Firstly, the DNA is going to be isolated. It is going to be sequenced. From that we can identify the VNTR sequences, right? Because of course in its DNA also the VNTR sequences are going to be present, and that VNTR sequence is going to be taken up, and against that we will be making probes. So let's suppose if from the crime site we have identified a VNTR, right now I am taking this sequence only. Of course the repeat is higher, but I am taking it as 4, right? So let's suppose this is the VNTR that is being found in this particular DNA. So we will make a complementary DNA of it, and the sequence will now become this. It will be tagged with a radio label. So now this will be acting as a probe. So probe is basically a molecule that will help us to identify that where exactly this sequence is going to be present, right? So we have created this VNTR probe. Now let's suppose we are suspecting that this, the DNA sample that we have found is related to these individuals. Let's suppose individual number 1, individual number 2. We are suspecting that either of them are having this DNA sample on the crime site, right? So what we will do, we will take the DNA from these 2 individuals also, separately cleave them with restriction enzymes, and then separate it on a gel, electrophoretic gel, right? So if I take it and separate it on an electrophoretic gel, if we separate it on an electrophoretic gel, so we will do the random analysis like this. So let's suppose this is the DNA from crime site. So I'm writing it as DNA C. Let's suppose these are the band patterns that we have found out, right? Then we are taking it from the individual 1 and individual 2 also, and we're going to segregate it like this also, right? तो हमें random bands दिखेंगे, ठीक है? Now we have to see that which particular individual 1 or 2 has that particular VNTR sequence that we have caught it from the साइट. सो पहली बात तो वो इलेक्ट्रोफोरेटिक जेल पे हम एनालाइज नहीं कर सकते, राइट, क्योंकि इलेक्ट्रोफोरेटिक जेल इज बल्की एंड द एनालिसिस विल बिकम डिफिकल्ट. ऑल्सो दैट प्रोब दैट इज स्पेसिफिक फॉर the sequence that we need to identify, वो भी इस पर bind नहीं कर सकती. That is the reason why we go for another process, the fourth step, and that is southern blotting. So in blotting what we do, we'll take the electrophoretic gel, then we will be putting the nitrocellulose membrane over it and then put certain weights over it, right. This is how what will happen, the DNA that is being found in the gel is going to transfuse into the membrane, right. So this membrane is too thin and this membrane is having the optimum width for identifying the bands that are being present, right. So after this southern blotting is being done, the DNA samples have been transferred to this particular nitrocellulose membrane. Then what we will do? Then we'll do hybridization with VNTR probes. Right? We'll do the hybridization with VNTR probes. Now because VNTR probes are specific, right? वो हर किसी में present नहीं होता. पहली बात तो same sequence. उसकी मैचिंग ही तब होगी अगर मान लो आपकी एग्जैक्ट मैचिंग हो रही है, मतलब डीएनए सैंपल हैज़ बीन टेकन फ्रॉम द सेम ऑर्गेनिज्म, राइट. तो पहली बात तो क्योंकि वो सीक्वेंस स्पेसिफिक है, फिर वो रिपीट नंबर इज स्पेसिफिक, तो इस वजह से कभी भी ऐसा नहीं होगा दैट द VNTR probe is going to bind to somebody who is totally unrelated, right. So after the hybridization is being done, now is the time to analyze it, and we analyze it with autoradiography. Autoradiography helps to identify all the places where the radioactivity can be seen, because these VNTR probes are radioactively labeled. So with the help of autoradiography, we could observe that where exactly the binding has been taken place. So let's suppose this is the nitrocellulose membrane, right, and these two are the individuals, individual number 1 and individual number 2, let's suppose, and this is again the DNA that has been taken from the crime site, right. Let's suppose this is having the sequence like this. So we cannot see the bands as such. So now when the VNTR probes has been hybridized, so because we have created it according to the DNA from the crime site, so over here complete binding will take place. Here let's suppose we are having this much amount of binding. Here let's suppose only one binding take place. So we can clearly say that individual 1 is the one that has left the that particular DNA sample at the crime site. So this is more related. Similar way we can use it for paternity testing. So let's suppose if it's the DNA from the child, so individual 1 is more likely to be the father of this particular child, because as I've told you that polymorphism is inheritable. It is transferring from one organism to the other, and it is inheriting from one generation to the other. So most probably this is more likely to be the father of this particular child in comparison to individual number 2. So this is how we actually check out that how the DNA samples that has been taken from different sites are being related to certain individuals or not. So this particular technique of DNA fingerprinting is being used for paternity testing, even to solve the crime disputes, right, and many more such sort of cases we can use DNA fingerprinting for. So this particular chapter is really important because there are many topics, very important ones, and every year multiple questions also came up. Like I have took certain questions from the year 2023, like this particular question which says that name the component that binds to the operator region of an operon and prevents RNA polymerase from transcribing the operon, right. So this is related to Lac Operon, and we know that the component that binds to the operator region of an operon is the repressor protein, and this won't allow the RNA polymerase to bind at that particular site so that it could transcribe further. If we talk about the inducer, it's incorrect because inducer binds with the repressor protein, not with the operator region. Same goes for promoter. Promoter is present adjacent to the operator, and promoter gives the site to the RNA polymerase to bind. Now regulator protein can be ruled out because over here when we talk about protein, regulator protein, right? So regulator protein could be of any nature. It's not necessarily to be a repressor. So regulator protein can be activator also, right? So that is why this is not being considered as a correct option. This one will, right? Another question that we are taking is the last chromosome sequence in Human Genome Project was, as I've told you, the last chromosome whose sequence has been completed in May 2006 was chromosome number 1, right? So all other options can be ruled out because this is a simple fact-based question, right? Another question we have is that there are two statements that's been given. The process of copying genetic information from one strand of the DNA into RNA is termed as transcription. So this is correct, right? Only one strand of DNA will act as a template, and against that we are going to produce the RNA. So that particular process is known as transcription, right? Then statement 2 is saying that a transcription unit in DNA is defined primarily by 3 regions in the DNA, that is promoter, structural gene and terminator. So this is also true, right? जब हम transcription unit की बात करते हैं, 3 regions are specific. One is promoter, that is the binding site for RNA polymerase, then structural genes against which we are going to form the RNA, and then finally terminator where the transcription is going to get ended. So this statement also being true, the correct explanation is being present in option number 1, which says statement 1 and statement 2 both are true, right? So this is how these kind of questions comes from the chapter, and every year multiple questions have been repeated. So please pay attention while reading this particular chapter, try to analyze it more and practice as many questions as you can and stick to NCERT. So thank you very much for attending this session. Hope you understood it well. Thank you very much.